Ramit, please start the IVRA song.
जय जय कृषि परिषद भारत की, सुखद प्रतीक हरित भारत की। कृषि धन पशुधन मानव जीवन, दुग्ध मत्स्य फल यंत्र सुवर्धन। वैज्ञानिक विधि नव तकनीके, पारिस्थितिकी का संरक्षण। शस्य श्यामला छवि भारत की, जय जय कृषि परिषद भारत की। हिम प्रदेश से सागर तट तक, मरुधरती से पूर्वोत्तर तक। हर पथ पर है मित्र कृषक की, शिक्षा शोध प्रसार सकल तक। आशा स्वावलंबित भारत की, जय जय कृषि परिषद भारत की। जय जय कृषि परिषद भारत की। मानव प्रिय कृषि सहचर हितरत, जय हो पशु संस्थान। भारत के कोने कोने तक पहुँचा नव विज्ञान। 19वीं सदी प्रथमोदय, पूना मुक्तेश्वर नव आलय। इज्जत नगर बना मुख्यालय, अभिनव अनुसंधान। बना विश्वविद्यालय, शिक्षण शोध चिकित्सा पशु संवर्धन। ग्राम अभ्युदय श्वेत क्रांति, सहयोगी शुभ वरदान। जय हो पशु संस्थान। विविध व्याधि के टीके निर्मित, रोग अनेक हुए उन्मूलित। वृंदावनी सदृश गौ विकसित, जन पोषक अभियान। मानव प्रिय कृषि सहचर हितरत, जय हो पशु संस्थान।
Thank you. Extending our heartfelt
इसका मेल नहीं गया ना, मुझे पता नहीं था इसके बारे में।
sir to felicitate Madam Dr. Roopsi Tiwari, Joint Director Extension Education, with the floral bouquet. Thank you, sir, for doing the honor. May I now request Dr. Arnab Sain, sir, to grace the event with the welcome address for setting the tone of this event.
Thank you, Dr. Bablu Kumar. Most respected and honored Vice Chancellor and Director IVRI, Dr. Triveni Dutt sir. Most respected Dr. Prabodh Bora sir, Director of Research, Assam Agriculture University, Khanapara. Most respected Dr. Roopsi Tiwari ma'am, Joint Director, Extension Education, IVRI Azatnagar. Respected Dr. Burman sir, the coordinator of the One Health platform for the Northeast and Professor and Head Microbiology. my colleague Dr. Pramod Dhar sir, Dr. P.S. Banerjee sir, Dr. Y.P. Nanda, Dr. Anuj, Dr. Bablu Kumar, and we have a very bright young biotechnologist, Dr. Rupam Datta from AU Khanapara, all state directors. It's a very momentous occasion for us today because this is the first time I think possibly that all of us are together under one platform in form of an interface meeting, and I am very thankful to our honorable director sir for having conceived the entire idea of working closely with the northeastern states and bringing in issues of significance on a platform so that we can strategize and plan the future course of action in terms of deployment of IVRI specific technologies, in terms of enabling training, because there are lots of value added like modules and skill development programs being run at IVRI, which could definitely be regularly conducted in various states of Northeast. So that's the very purpose of conducting this first meeting, and I'm sure that there will be many more in the days to come. And I sincerely thank all of you from the core of my heart for having accepted our invitation, because unfortunately we had to once postpone this event by a week or so. But despite this, all of you coming here today means a lot, and I really welcome you to this particular event and look forward to a very active, fruitful, and productive collaboration in the days ahead. Thank you.
Thank you, sir, for a warm and insightful welcome address. Your words have set the tone for this gathering. I now kindly request Dr. Ruth Tiwari Madam to provide a brief overview of the program and enlightening us on the main agenda.
Thank you, Dr. Basu. A very good morning to you all. Most respected Dr. Jainat sir, Honorable Director and Vice Chancellor of IVRI, Dr. Prabodh Bora, Director of Research, Assam Agriculture University, Khanapara, Professor A. Burman, Coordinator One Health Program for the Northeast Region, AU Khanapara, the representatives of the Animal Husbandry Departments of various states, Dr. P.R. Joshi, Joint Director, State Veterinary Council, Meghalaya, Dr. H.P. Paul, Deputy Director, Administration, Meghalaya, Dr. D. Ganguli, Director, Animal Husbandry Department, Arunachal Pradesh, Dr. M. Taipo Sia, Director, Animal Husbandry, Arunachal Pradesh, Dr. Kardom Basar, Deputy Director, Animal Husbandry, Arunachal Pradesh, Dr. Seva Yamro, Senior Veterinary Officer, Arunachal Pradesh, Dr. Sanjay M. Gajmer, Director, Sikkim, Animal Husbandry Department, Sikkim, Dr. Krishna Rao, Veterinary Surgeon, Nagaland, Dr. Keduzul, Deputy Director, Nagaland, Dr. C. Raudrint, Director, Animal Husbandry, Mizoram, Dr. Muti Rahman, Joint Director, Institute of Veterinary Biological, Assam, Dr. Vipul Nath, Veterinary Officer, Khanapara, Assam. These are the representatives we have here today in the meeting. I think the Tripura representatives from the Tripura Animal Husbandry Department, they will be joining shortly. And from the Manipur, I think they have joined virtually. Apart from that, we have our joint directors joined from the IVRI Isanagar, Dr. S.K. Medirathan, Joint Director Academic, Dr. Pallav Chaudhary, Joint Director Bengaluru Campus, Dr. Biblendu Mandal, Joint Director Mukteshwar, Dr. Gorakhpur, Station in Charge, IVRI Regional Station Palampur, Dr. Arnab Singh, Station in Charge, IVRI ERS Kolkata, and coordinating the whole program, Dr. Athel, Station in Charge, Tech Pune, Dr. P. Dhar, Head Standardization Division, Dr. P.S. Banerjee, Dr. P.K. Nanda, Dr. Anand Chauhan, Dr. Bablu, senior officials of the State Department of Animal Husbandry of the Northeastern region, and the chairman, co-chairman, rapporteur, speaker, and the coordinators of technical session and faculty and students of IVRI who have joined virtually. Warm welcome to this interface meeting series. Actually, it's a series of meetings we have been organizing with various states of the country, and in this row we have organized. This is the fourth meeting in the series. Earlier to this, we organized with two states, Maharashtra and Himachal Pradesh, and then we went into organizing one All India State Department of Animal Husbandry meet. Maybe some of you may have joined it. It was in a virtual mode, and it was throughout the country. But in that meet, we felt that there was limited scope of interaction with the officials of the Northeastern region, and therefore, keeping in view the different needs that arise from that meet, we felt that there was a need to organize a full-fledged meeting for the Northeastern region. And because this particular region is quite different from the whole of the country with 7 sister and 1 brother states, and a population of over 45 million, which is around 3.76% of the country, and the livestock population is 4.54% of the total national livestock population. This particular area is home to many unique breeds and livestock species, but the animal husbandry practices, they are perceiving many challenges like the porous international boundaries, the transboundary emerging diseases, emergency selling of products, climate changes, and recently the devastating transboundary disease, the ASF in pigs that caused huge mortality and economic loss. So to have a better understanding of the problems of the animal husbandry in this particular region, this meeting has been planned. It's in hybrid mode so that a number of officials of IDRI and head of the divisions and all of them, they can provide their input. One of the major reason this meeting was organized was that that the regional committee meeting 3. The committee 3 has all the northeastern states in it, and they provided a recommendation in that repeatedly for the Mizoram state especially, that is establishment of the biological product unit in that, and the capacity building of the officials of the State Department of Animal Husbandry of Mizoram. We could not complete this particular target of the regional committee 3 recommendations because of maybe we have repeatedly written to the state, but I think this meeting will provide us a good platform to fulfill this particular requirements of the regional committee 3, and we can take some initiatives together here. Apart from these, the specific agenda and the major focus is to foster more collaborations, share knowledge, and discuss what are the latest advances that we have developed at our institute that hold the potential to revolutionize the livestock and development in this particular area. And we aim to bridge a strong gap and between the research and the development of the institute and the practical needs of this particular region. So we also aim to acquaint the directors and the officials of this state animal husbandry department of the any region with whatever developments we have taken up, the cutting edge innovations and breakthroughs which our institute has achieved. Furthermore, we are seeking to create some partnerships and promote joint research initiatives and facilitate the growth of entrepreneurs and startups, develop collaborative training programs for various stakeholders, and develop a long-lasting license for improving the production and productivity of livestock sector in this region. We recently had one training of the northeastern region, and I found that we had around total 10 participants in that. It was in the online mode. We organized it in the online because physical mode much participation was not there, but in online there were 10 participants from few states, including Mizoram. So at least that was fulfilled, but it was held very recently, I think around 10 days back. So I think today's discussion it will provide us effective insights into the performance of the technologies at the field level and what constraints you are facing by using the animal science technologies. This we can identify sitting together here. So additionally, we will deliberate on what are the emerging issues and problems that require urgent research and intervention in the NE region. By coming together in this interface meeting, we hope to identify researchable issues that are aligned with the evolving needs of the livestock sector in the NE region. I once again extend a very heartfelt welcome to one and all. We look forward to very fruitful insights and deliberations in the meeting for a bright future of the livestock sector in the NE region. Thank you, Jai Hind, Jai Bharat. So before moving further, let's look at the expectations of the State Department of Animal Husbandry of the Northeast region. So we have asked you to fill a Google form, and through that we found some of the expectations and the needs which you have filled up in the Google form. May I have the slides, please? Next slide, please. Next slide, please. Yeah, so this particular meeting is unique in the nature that we have for the first time organizing an interface meet at the north, including all the states of the northeastern region. Now, earlier in the yesteryears, in around 2013, 14, and 16, we had organized a meet specifically with certain states, but not together the whole NE region. So this is a unique opportunity and a unique platform indeed where we can have the sharing of the good practices across states in these states. So we have the registrations received from all of these states. Next slide, please. What specific assistance you expect from IDRI? So certain assistance, of course, ideally may not be into because it's a policy decision like establishment of ICR Nivedi Regional Center at Umiam and College of Veterinary Science in Kadarmulai. And then training and capacity building of veterinarians. Of course, this is one of the major agenda to take up today, how to develop these collaborative training programs, wherein within less time and less of the you can say time consumption and other things, we can organize these programs without sparing your because the manpower is very essential in the state departments, and how in the less time we can do these trainings. Policy support and advocacy in conservation of different breeds of livestock, research and development in fodder production and nutrition, area specific collaborative projects. Of course, this is one area we would like to associate with animal disease diagnosis and diagnostic kits, quality veterinary biologicals and quality control of vaccine, and scientific prophylactic advice regarding disease outbreak at endemic area. Next slide, please.
Slides are not visible, Shanti madam.
Slides are not visible.
Hi.
Is it visible, sir, now?
No.
No. Are we sharing this slide, please?
Yes, yes.
Share from
We are stopping sharing.
now you share.
Is it visible now?
No. Please share presentation. We are seeing the screen where the folder is visible for us.
Sometimes because of this issue.
You open the presentation.
PowerPoint
Only
खोल के
share
रखिए।
the whole screen.
एक window में।
Screen.
Yes.
PowerPoint खोल के रखिए।
Share entire screen, not the presentation.
Yes.
First you open the presentation.
Open it.
Share entire screen.
अभी नहीं stop से
Interface meeting है ना.
minimize it. No. Open the PowerPoint. Now we go back to that meeting. Share it.
No, entire screen.
Share
From
screen.
upcoming videos.
Share enter screen options. Okay.
Now it is.
No, it must be visible.
Is it visible?
Yeah.
Now it's visible now.
Yes.
Okay.
Is it visible, sir?
Yes, it is.
Yeah. Okay, thank you. Next slide, please. So, Sriwali, back to you.
One slide back.
Yeah. This is what they, in the Google registration form, the states have expected from IVRI. Establishment of ICR Nivedi Regional Center at Umiam and College of Veterinary Science in Kadarmulai. Of course, this is not in the purview of IVRI, but of course we can discuss at some policy decisions we can take up with the ICR. Then training and capacity building of veterinarians. This is one of the important agenda of today's meeting itself, that how we can develop a collaborative training programs with all the different states and how these can be taken up, whether they should be in the online mode or in the physical mode, how much they can be in the online and offline mode, that has to be decided. Then policy support and advocacy in conservation of different breeds of livestock. Then research and development in fodder production and nutrition. Area specific collaborative projects, of course this can be taken up together here. Animal disease diagnosis and diagnostic kits. We have many diagnostic kits that will be shown by one presentation by Dr. Anuj. Then quality veterinary biologicals and quality control of vaccine. So Dr. Dhar, he will be putting the presentation on the development of the veterinary biologicals and the testing also. Scientific prophylactic advice regarding disease outbreak at the endemic areas. Next slide, please. These are the areas in which veterinarians of the state require training. So practices involved in conservation of indigenous breed of livestock and poultry, state-wise epidemiological surveillance of notifiable diseases, advanced disease diagnostic techniques, animal nutrition related advanced lab techniques, innovations and new treatment protocols standardized by IVRA, orthopedic techniques in large and small animals, training in ophthalmology, echocardiography and ultrasound in small and large animals. Next slide, please. Areas in which paravet staff of the state require training. So mapping of areas where a specific disease is prevalent, animal restraint, type of fodder grasses and its propagation, recording clinical data of the patient, artificial insemination and assistance during ETT, innovations in animal husbandry practices and extension activities, practical courses in handling specimen collection, packaging and transportation, proper AI with sex-sorted semen and steps to be followed in AI, diagnostic techniques and surveillance of new emerging animal diseases. Technology requirements for veterinary officers. Infertility and mastitis management, AI in equines. Procedure and interpretation of imaging technologies, there is X-ray and ultrasonography. Vaccine production, particularly cell culture, anti-rabies vaccine. Checking of feed samples and its legal aspect. Disease diagnosis and disease control. Prevention control of transboundary diseases. PCR techniques for animal disease diagnosis. These were the requirements of veterinary officer. Now the technology requirements for farmers as suggested by the state representatives of the Animal Husbandry Department. Economic farm management, remote veterinary consultation services, fodder and nutrition management tool. Like remote veterinary consultation services, we do have an online veterinary clinic app, so that can be used for that. Fodder and nutrition management tool, livestock handling techniques, livestock products marketing, farm economic and record keeping, app-based information systems for knowledge sharing. We do have many apps like that, so we'll be telling about that. Prevention of infectious diseases. Next slide, please. Would you like to suggest any researchable issues in animal health sector to be undertaken? So we've got these responses. Research on African swine fever with special reference to vaccine production and effective prophylactic measures. Thermostable vaccines. We do have developed some things in this area that we'll be sharing. Then proper use of powder and urine in making and marketing products like funeral pyres, flower pots, etc. Documentation, research on A2 milk of Siri cow. Device and methodologies to reduce the nuances of street dogs. Study and research of decreasing the population of monkey. You can see the last one, please. Take it above. Conservation and propagation of indigenous pig, poultry, cattle, and goat breed. So these were the areas that have been suggested by the representatives of the State Department. So with this, thank you very much. I think this presentation is a platform to set up the page for the meeting, and I think we can now delve deeper into after this basic presentation. Thank you.
Thank you, madam, for providing us with the comprehensive overview of the program and setting up the tone of this meeting. We always appreciate your effort in and guiding us in organizing such events. May I now request our Honorable Director from Vice Chancellor IVRI, Dr. Surendra sir, to share your valuable remark with us, sir.
Thank you very much, Dr. Kogu. A very good morning to all of you. Esteemed colleague, Dr. Director of Research, AU Kolkata. Colleague Dr. Ruchi Tiwari, Joint Director of Education, IBRI. Dr. Anand Arman, the coordinator of this program of NERD. Esteemed colleagues from IVRI, Dr. Arnav Sen, in charge and head of legal sector, ERS, IVRI, Belgacia. Dr. Banerjee, principal scientist, and our senior learned colleague at ERS, Belgacia, Dr. Nanda. And we have the head standardization division, Dr. Peter. Our esteemed guests from various important states of the country, dear directors, really it is a matter of great pleasure that IVRI along with the Khanapara, they are meeting with our senior colleagues, director colleagues working across various states of northeastern states. As we are aware, the focus of Government of India and also the stress given the R&D, infrastructure development, quality control development of these north states, we know. So, in tune of the focus of Government of India, especially Honorable Minister of Agriculture, the important points emerged out of the meetings of Honorable Agriculture Minister's review meeting and also the regional committee meeting of northeastern states. As shared by the learned colleague at Rusi Tiwari, that one important aspect is the handholding of establishment of biological production unit. Various problems faced by northeastern state related to livestock health, and also the capacity buildings of our state officials working in the field or handling the eventualities. As we are aware that the status of livestock sector in the north eastern region, the cattle, buffalo, sheep, goat, pig, and horses and ponies, the livestock population is reducing drastically in these states, the tune of 8 to 48%. So this is our concern. This population is reducing drastically in these states. And you see our new donkey and camel population also reduced up to 75% plus. And certainly this yak, mithun, and the poultry population structure is going up. And we are also aware that the loss incurred due to the various incidents of diseases. As a whole, in the country, this loss is in the tune of rupees 140 to 200 billion per year. And we are also aware this in cattle, in bovine mastitis rates first, giving loss to the country, and out of this loss, 70% subclinical mastitis, which we are always ignoring. We are also know that this the livestock population structure, northeastern states, they have the 4 to 6% of the total cattle, sheep, and goat of the country, and we have a rich in pig population now. There is a setback to this industry. We are having 40% of the total veterinary population existing in the state. And we have AR 23% of the total country's population and 11%. But we have lot of strength. Along with we have certain weaknesses in these states. Keeping in view of the strength, weaknesses, and we have a number of opportunities also. You see, if we see the climatic conditions of this whole region, very you see high rainfall and humidity. It is really a good, very good environment for pathogens, maintenance of pathogens, and also the dissemination of it. We also know there is a very close interactions among human beings, domestic animals, and wild animals in these states. We also know that a 4,500 kilometers, you see international boundaries along with these states. So unrestricted movement of the animals is also a great source of various transboundary animal diseases. So these are the various, you see, weaknesses, or we can say the negative points of factor which we are having in the northern states. So now the keeping in view of the strength, weaknesses, opportunity, and the needs of northeastern regions, we analyze what would be the way for improving the livestock production and productivity of states. The strengthening of infrastructure is one of the important you see component. Although we have a very good infrastructure here in terms of human resources and also in terms of various institutes and laboratories, but still we are lacking the need to strengthen the infrastructure for surveillance and monitoring of various diseases for early warning. This early diagnosis of various, you see, emerging infectious diseases, exotic diseases, and transboundary animal diseases are utmost important. This is one of the very important, you see, activities to be focused in these states, and also the establishment of appropriate and adequate you see quantity of this biological producing of biologicals through good biological units. We have number of institutes, ICR institutes, and also the various government of India's initiatives, and also the institute governed by government of India, located at northeastern states. But we find that the networking are really required. The networking of the institutes established by the state government of India in this region, in this northeastern state, is the networking among the institute is really required. And the link with the referral institutes, national institutes linkage is also we are finding you see peak. So the collaborations among various states with the national institutes is also important to address the various issues, emerging issues in these states. And finally, we really required to create the pool of trained human resources here at all our 8 northeastern states. And so the creation of the pool of trained manpower is very important in order to handle the problems or various eventualities occurred at various point of time. So you see, the capacity building of our human resources working in the field is very important. So today's meeting, the agenda is as per the expectations of the government, there is a need to establish a biological production unit at the appropriate place in order to cater the need of various vaccines. And also the collaborations with the various states in the area of various you see emerging issues. So the research collaborations is also very important. And third very important is the creation of trained human resources. What we have observed that we have a very good infrastructure at our AU Khanapara, and we have a memorandum of understanding for research, teaching, and various outreach activities with AU Khanapara. And what mechanism we can develop in some areas of research, you can have a greater collaboration with the AU Khanapara. And also in various areas of capacity building programs, we can also have the collaboration with the AU Khanapara. Because IBRA is a 135 years old institute. We have a really good human resource pool, good infrastructure which we have developed over the period. And IBRI is you see, because of the IBRI, we have reduced drastically animal sufferings due to various diseases. Fortunately, whatever the vaccines used in our national animal disease programs have been developed by IDRI, and diagnostics also have been developed by IDRI. Currently, you see, we have a colleague, Dr. Anuj, he will be presenting the research technology portfolio of IDRI. But in brief, around 44 technologies, including 11 vaccines, we have commercialized with 156 commercial houses. We have also developed a total of 44 ICT tools, which are being used in various 134 countries with a 5 lakhs plus our stakeholders. So these ICT tools we have already developed, which we have seen the requirement of NEA. So you see that we must start the greater collaborations with the national institutes. Then we can initiate or introduce some of our good R&D activities, and also the outreach activities. Outreach activities we are already doing through our ERS Valencia, but various you see R&D and training activities are really required. So what today we would like to discuss that we have already seen, we have already looked the expectations of our NES, Northeastern State, the IVRI, and we know the strength of the IVRI, and recently in tune of national education policy, we have introduced 91 various national diploma certificate courses and our vocational education and training courses. So these, because you see the government of India initiatives that whatever the infrastructure in terms of manpower and laboratories established at IBRI, this should be opened up with our various stakeholders. So, in tune of National Education Policy 2020, we have introduced 91 various diploma courses and training courses and capsule-oriented courses of the in expectation of the various states. So, what I have observed in the presentations, the expectations of our states located at Northeast, all the capsule-oriented courses we have already developed. So there is need to have a connect with the IVRI, and I think due to shortage of manpower, state may not be able to spare their human resources for a long period. So the courses of long period can be, we are seriously thinking to implement through our online mode. So our veterinary officials working in the field can be can updated their knowledge. And some of the important courses of you see 3 to 7 days capsule-oriented courses, which are really required some hands-on trainings in the laboratories or you see in livestock farm. So would like to have these capsule-oriented courses in offline mode. And we are seriously also thinking, and we will discuss with the in Khanapara that some of the courses which are really practical exposure can be given at Khanapara, and these courses can be run in hybrid mode. Various theoretical classes can be completed through online mode, and practical classes can be completed through offline mode. And if required, some of the expert from IBRI can also be deployed for sometimes during the training period at AU Khanapara. So these type of various mechanisms can be developed in order to strengthen the linkages with the northeastern state. So I think we have the strength, and we have livestock wealth and diversity and various states. And we have created a huge infrastructure as AU Khanapara also. And as you are aware that this is strength of idea. So I think we have to work very seriously as per the expectations of Government of India and expectations of our stakeholders. You see, nowadays the entrepreneurship development and the startup development have become a very important for the states as well as for the students. So we are also having very, rich ecosystem in order to develop startup and entrepreneurs at IBRI. Government of India have also given the responsibility to initiate a RAVI programs, Raftar Agri-Business Incubation programs, with financial support to nurses, to entrepreneurs and startups. And we are very happy that we have some of the startup on roll located in these states. So due to some unforeseen situations to outbreak of ERS, some of the startups they really collapsed. They could not proceed. But I think we have a strength in these states. We have a great livestock resources and the diversity. We can initiate some of the good collaborative R&D and our capacity building activities in order to serve the livestock owner in a very effectively. So I think today we are here to listen our learned colleagues, director colleagues of various states. So based on their discussions, we can set our researchable agenda and also the future collaborative programs with IBRI and also along with AU Khanapara. So these are the main focus today's meeting, and I am really grateful to all our esteemed directors who have spared their valuable time out of their leadership. I know the working of the state department of animal husbandry. You are very busy in handling the state issues. You spared your valuable time. We are extremely grateful. I and on behalf of IBRI fraternity, we welcome you again and with the expectation that we will have a better collaboration. We are also extremely grateful to our learned colleague Director of Research of the EU Paratana, Dr. Umar, who have also studied at this time. And we know that he is very generous, very humble, and I think we will have a good time ahead in order to have a good collaboration in future ahead. And thanks to Dr. Varman, and he also, I think, he has a very important program on this state. And we will have also, you see, potential how best we can collaborate various R&D activities. And in the end, we are also grateful to our colleagues working in, you see, working with very sincerity with the Northeast state, our colleagues at ERS Valdezia, ERS Highway Valdezia. Head regional center, Dr. Arabsen and Dr. Banerjee and Dr. So with this small view, we will discuss and we'll have a great discussion. We'll have a with the expectation we'll have a great conversation in future in order to solve the large task. So, thank you. Thank you.
Thank you very much, sir, for your inspiring word and apprising briefly about the offerings and the technology backstopping that IBRI can provide to the Animal Husbandry Department of Northeastern State. You very briefly narrated also the strength and weakness of this area, and you raised your genuine concern about the rising transboundary diseases. And you suggested two very important suggestions like strengthening infrastructure, setting up a biological products unit, better networking for better collaboration and convergence, and creating a pool of trained human resources. And you also narrated the expectation from the deliberations of today's meeting, sir. So thank you very much. So before we proceed, may I now request all the participants to kindly introduce themselves one by one so that we can know each other. We can start from one side, sir.
Good morning to everyone. I am N.N. Varman, Professor and Head Department of Microbiology. So, I'm N.N. Varman, Professor and Head Department of Microbiology, College of Life Science.
Good morning, I'm Arnab Seth, Head Regional Center, here in Kolkata. I was very closely associated with Northeast. I spent 12 years clearly in Mubium and Manipur. Thank you.
नमस्कार, मैं डॉक्टर PS बैनर्जी, प्रधान वैज्ञानिक, भारतीय पुष्टि अनुसंधान संस्थान पूर्वांचल केंद्र।
Good morning. Myself Dr. Kovita Dasgupta, Joint Director ARTD, Government of Tripura.
Good morning, everybody. I am Dr. Sanjay Zatkar. I represent Sikkim. I am the Director for Extension and Training, Cattle and Dairy Sector.
Good morning, everyone. I am Dr. Rutu, Deputy Director, representing Nagaland.
Good morning, everyone. I'm Dr. Rexo. I'm from Nagaland. I'm working in the state license.
Okay. Myself, Dr. Cyril Rintuanga, they call me Rina. I think most of you might have recollected our days. I'm Director Age and Mediation Government of Mizoram.
Good morning. I'm Dr. P.R. Joshi. I'm Joint Director in Government of Meghalaya. I'm also Registrar of Meghalaya State Veterinary Council. Thank you.
Good morning. I'm Dr. H.P. Paul. I'm the Deputy Director Administration. That was in the Directorate Animal Husbandry and Veterinary.
Good morning. I am Dr. Anuj Chauhan, And I'm looking after ITMU and swine production farm at ITMU.
Myself, Dr. Bablu Kumar, Principal Scientist. I am posted in the Biological Products Unit of the IVRI, and I am also looking after the entrepreneurship and startup activity at IVRI. Thank you.
Very good morning to one and all. Myself, Dr. P.K. Nanda. I am working as a Principal Scientist at ERS Kolkata Center of IVRI. Thank you.
Good morning, everyone. Myself, Dr. Rupam Datta. I'm Assistant Professor, CBSE Khanapur.
Good morning, everyone. I'm Dr. Seva Yandu, Senior Veterinary Officer, Department of Animal Husbandry Veterinary, Government University.
Good morning to all. I am Dr. Karman Basar, Deputy Director from our hospital.
Good morning, I'm Dr. Saikodia, representing for Mexico. Namaskar and good morning to all. I am Dr. Ranjan Nandi.
Good morning, everyone. I am Dr. Gayatri Das, Veterinary Officer working at the Institute of Veterinary Biologicals, Kanapura, Kolkata.
Good morning to all. Myself, Dr. Vipulnath, Veterinary Officer, Institute of Veterinary Biologicals.
I am Principal Scientist and we are using the Biological Simulation of ERS.
Good morning. I'm Dr. Prabhu Bora, Director of Research Veterinary, Assam University, Khanapura.
Good morning, I am Dr. Rupsi Tiwari, Journalistic Station Education, Library ICT.
So, may I request the director from Environment Department, Manipur?
Good morning. I'm Dr. N.G. Wattubi Singh, Director Veterinary Department of Manipur. Good morning, everybody.
I kindly request anyone else who is present online from Animal Husbandry Department may kindly introduce yourself. So we have reached to the end of this inaugural session. Now we move ahead to the technical session 1, which will be chaired by Dr. Arnav Singh sir, co-chaired by Dr. Peter. Dr. Akhilesh Kumar will be, and he is the coordinator of this. Dr. Anuj and Dr. M. Kumar is the reporter of this session. Now I request the chairman of the session to continue the further proceeding of this session 1.
Thank you, Dr. Bablu, with permission of the chairman. Now request the first speaker, because we have basically 3 talks in this session. So the first speaker, Dr. Pranav, will be speaking on the production and quality control of veterinary biologicals. So may I now request Dr. Pranav to kindly start his deliberation, and I request Dr. Rupam to coordinate with the slides. Thank you.
F5.
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 Stop share. Make it full screen, then share. Stop share first.
Stop share.
Then you just reduce that and open it full screen. Yeah. Now go to full screen mode. Yeah.
Share the
Yeah. Let's share.
Share the entire screen.
Screen, screen.
Always share the entire screen. Then only.
Is it coming? It's not coming. It's coming.
It's coming. Sir, are the slides visible to online participants? Anybody can from IBRI can respond. Are the slides visible?
Yes, yes, visible.
Then you can go like this.
Yes.
It is not in the slide mode, not in presentation mode.
Now,
Yes, it is on presentation mode now. In here. Yes, it is visible. Okay.
Dr. Burman, Head of the Department of Microbiology in Kolkata, Dr. Arvind Sen, Dr. Banerjee sir, my other colleagues, all the dignitaries from different states of Northeast, and all my joint directors and HODs and heads of regional centers.
Excuse me, sir. Sir, your voice is not clear.
Voice? Okay. So today's agenda was the establishment of biological unit at Khanapara. So I will do my discuss my slides in that mode. So before I start, that this title is Production and Quality Control of Veterinary Vaccines. On the top, you can see that common seeds for which vaccines are made in different parts of the country. Like I'll start from the left, is NDV, PTR, IBD, then IB, rabies, Meniere's disease, duck plague, classical swine fever, then LSG, Sigfox, Pox, and Fowl. These are the viral vaccines. And among the bacterial vaccines which have been manufactured in different state biologicals or companies are mostly HS, fowl cholera, then brucellosis, anthrax, and etc. Next slide, please. So when you talk about veterinary biologicals, I'll start with this first slide. First slide, you should have a good recognized facility. That is a GMP. You can go to that number 6, it is not visible from here. Number 6 says you should have a test license for manufacturing the vaccines under Form 28D. You can go to this CDSCO. CDSCO stands for Center for Drug Standard Control Organization. This is the actually regulatory authority which provides license. So before you think of producing any vaccine, first you have to take license of the vaccine, and under that license, you have to have that GMP facility. Once you apply for the license, they will ask whether you have a GMP facility. Once you develop the GMP facility, you have to again report to the CDSCO that yes, you have a GMP facility. CDSCO will make a joint inspection with the drugs authority of local as well as central, and they will inspect whether you are eligible for producing the vaccine or not. So that part, that is the first and foremost requirement. Then comes to you take the seed from any institute or any other source, and then you do some initial research, take a test license first, get these first 3 batches of those test licensed products from IBRI, and then apply for the complete licensing. Then again you have to produce 3 consecutive batches. Then DCGI will ask, will issue license subject to condition that 3 initial batches should be tested by IBRI. Then you get a complete full-fledged license, then you will produce and use for your state or other states. That is the procedure. Next slide, please. Before we start for any vaccine production, first is this vaccine seed. It is not visible here. It's vaccine seed. So seed you have to source from a reliable source, and it should be certified. it should be, all, everything should be known about the seed. Its titer, its cell count, if it is bacterial vaccine cell count, then number of vials you are sourcing, then all the SOPs, and all these requirements, which are regulations, everything should be in hand and should be certified by that, from where you are sourcing the seed. Next slide, please. Is not moving. Next slide. Yeah. It is a viral vaccine, cell culture vaccine. The first you have to see that cell health goods, you should have a very good cells. That should be, second point is very, very important. Cells should be free from mycoplasma. There are methods of removing mycoplasma from cells if it gets contaminated. There are some easy method for detection also. Then the good cell morphology is very much first requirement for any cell culture vaccine. And other prior preservations and all these procedures should be thorough. Next slide, please. These are the slide. These are the some simple techniques to detect mycoplasma. On the right is the PCR method. Single line means these are negative. If double band means the top one is the mycoplasma contaminated. And another method is detection by DAPI method. In the center, you can see some big dots and some small dots. Small dots, big dots are the nucleus of the cells. And small dots are the nucleus of the mycoplasma. If you detect, you can treat the mycoplasma removal agent, and finally you can get the left extreme left type of picture. There are only big dots, only nucleus, no mycoplasma nucleus. So then only your product will be good. You will have good titer. Because many times I attended many meetings on FMD, quality control meeting, PPR quality control meeting. The companies come with a complaint that they are not getting good titer, they are not, the vaccine getting failed. Then after meeting, we come down to that, narrow down to the one topic areas. Your cells are not good, cells are not healthy, cells look for mycoplasma. So I have made a specific one extra slide for this mycoplasma. Next slide, please. Then cell count and cell health and how much you have to optimize. Suppose you take a technology from anywhere, suppose classical swine fever, you should know what is the titer of the vaccine virus, what is the MOI, multiple infection, all this you should be knowing. Then only you can proceed for, then you can develop good vaccines. Next slide. Because the multiplicity of infection is very important. If you give, it should be a very optimal. If you give low, you will not get good titer. If it is high, this is happening in the manufacturing unit. You to get good titer, people are giving more virus. What will happen after 4 or 5 passages? You will get good titer, but all of a sudden you will not get titer. All of a sudden you will fall down. You will see that there is no virus. There is no virus. Thing is that DI particles will be produced, and you have to avoid having DI particle for that cell count, that optimization of MOI. These are all very much required before you go for the production. Next. And see, this is just to show that cell that stage is the initial stage. If cells have grown for 2-3 days, you should not infect. You should be very healthy. Infect the young healthy cells to infect with the virus for any vaccine production. Yes. Then I will talk on this seed lot system. Suppose any company takes some 4, 5, 6, or 10 vials from a source for vaccine production. Say for example, Assam, this state government biological units, they take a vaccine. They should go for seed lot. They should not immediately start producing vaccine from the initial source vials. One vial they should make a seed lot one. They may make 100 or 200 vials. Then they will test all the parameters as per Indian pharmacopoeia. Then once seed lot one is okay, then they go for one vial they will take, and they will take for seed lot two. Seed lot two again they will produce around 200, 400 batches, and then one batch they will test for everything, for those parameters. Then they can start producing seed lot two, every batch from the seed lot two. For example, suppose you get a vaccine at passage number 55. 55 seed lot becomes 56, seed lot 2 becomes 57. So your batch will be from passage 57, 58 like that, around the 5 passages you can go. And one thing you have to keep in mind that you should not give more passages. Suppose you have got a technology, you can go only up to 10 passages. So you have to plan like that. You should have enough seed lot 1, enough seed lot 2, so that your passage after 10 years, even 20 years, your passage should not go beyond 10 passages. Next slide. This is very important. I will talk in terms of I visited last year in Assam biological unit. The freeze-drying unit and freeze-drying unit is the finishing product you will get to the freeze-drying. After freeze-drying only you get finished product. The freeze drying, buying of the freeze drying is very, very important because it depends on the vaccine to vaccine. You should have one or two freeze drying, different types. Why? Because for freeze drying, you have to add some freeze drying solution. Freeze drying conditions will depend upon the solution. Suppose you are freeze drying something in skim milk powder. Their freeze drying condition, their mind temperature will be different from that vaccine which contains sucrose. There is a scientific term called eutectic point. You should know the eutectic point of all the ingredients you are using for the freeze drying. Suppose you are using sucrose or lactose for freeze drying. You should know what is the eutectic point of that ingredient, that is minus 30. So your freeze drying should have capacity to go beyond minus 30, 10 degree beyond, minus 40. You have purchased a vaccine, freeze drying for freeze drying of the bacterial vaccine, that will not work for viral vaccines. That concept I want to convey here. Because the Khana Dr. Roman may be knowing that they have purchased a freeze drying, that is a 41, and I just spoke to Dr. Motiram, Joint Director. They are going for a good one this time. Okay, next slide please. Then when you get the finished product, you should go through all the tests mentioned in the IP 2022. Like sterility, purity, safety, potency, extended agent, mycoplasma, and moisture. Each and everything is very important. Moisture, many times we omit moisture, but moisture is also very important because if the vaccine contains more than 3% moisture, it will not maintain the favorability titer for long. Immediately it will very quickly fall down. Titer will go down and vaccine will fail. Next slide please. Okay. Now previous slide. Now I will talk on the quality control testing of the different vaccines. Vaccines are livestock and food. We can quickly go through this. For quality control, I must say that if that product should be a well-researched product, well developed and field tested, and then it comes for the quality control. And quality control, we cannot do it haphazardly. It should be regulated. It should be as per the skin line, the approved method, and it should be harmonized everywhere. Next. Now, thing is, I told that quality control should be regulated. Who regulates? Who will regulate? There are two authorities, all guided by this Drug and Cosmetic Act and Rule 1940 and 1945. There are several schedules you can see. Most important here in today's meeting, today's topic is Schedule F. This vaccine and cell. All these monographs are given. How to test poultry vaccine, how to test swine fever vaccine, how to test all these things have been mentioned there. And those which are not mentioned there from 1955, pharmacopoeia came, Indian pharmacopoeia. Then after that, any updating, they updated in the pharmacopoeia. So now all the modules are available in the Indian pharmacopoeia. I will talk later. And regarding the GLP, Dr. Burman and Dr. Mathuraman, that GMP, it should be followed with that schedule name. Schedule name, it is written step by step. What should the class over class A, class B, class C, everything is written. What should the premises, what is the size of the premises, everything should be there. And one more thing, sir, they were showing concern that how to go for GMP. I would suggest that first authority should visit a company where they have GMP. First they will see their GMP and they can consult the local branch authority. Local licensing authorities are there. Every place is there. They can come, they can suggest this is wrong, this is good, this is not bad, like that they can suggest. And they can, and one more thing, third point is, you can hire a consultant, private consultants are there. You just Google search GMP experienced companies, you will get some number, maybe nearby from Kolkata to nearby. They will suggest, they will give a layout, doors should open which way, what should be the size of the room, what should which type of material should be used, which type of epoxy, which type of paint, how to be the corners. Suppose the drain, what's the room, how will be the trap, all different from our normal household things. So from there you get the idea. Then you have a plan, so it will be foolproof. Because when you say that, look, we have a GMP, these authorities will come from central, from state, and from IBM will come and will see whether it is misplaced or not. So better to handle with them and from the beginning. The first time foremost, you should go and see one company where they have GMP. That I can arrange. I visit for inspection. I can give the number and I can give the contact. Okay, next. Regulatory authorities are two actually in the country. One is IPC, all comes under DCPI. That's CBSE, Central Drugs Standard Control Organization. IPC, right side is the IPC, Indian Pharmaceutical Commission. They are the standard setting organization. They set the standard. What should the titer of this vaccine? What should be the titer? What should the expiry date? They only set the setting, set the standards. And CDSCO look for its implementation, whether it is being followed or not. They provide all this manufacturing license, any new drugs. They also done in the next slide. And if your vaccine contains any genetically modified agent, you have to take permission from DBT, like IBSC and RCGM permission and clearance on the GSE. Next slide. This is the, I will not spend much time. This is the CDSU, I already told. They approve new drugs, they give license, and even for amendment of drug and cosmetic acts and banning of drugs and testing of new drugs, this organization deals with all these things. Next slide, please. Yeah, now this is the CDL, Central Drugs Laboratory. There are for human vaccines, there are 6-7 CDLs. For veterinary vaccines, all the veterinary vaccines, IVR is the Central Drugs Laboratory. Well, of IVR as per some section laws, section 382. And Director IVR is the ex officio member of this DTAB. And Central Drugs Laboratory works on behalf of Director IVR. And all for testing of all the veterinary vaccines, serum, and diagnostics. And Director IVR is also the appellate authority for samples referred to honorable court. If any dispute is there, vaccine applied, animal diet, if there is any complaint, that will be tested by IVR and appellate authority is Director IVR. Next slide. I already told in India you can skip this Indian Pharmaceutical Commission. Yeah, it is not visible from here, sir. Pharmacopoeia started from 1955 and initially it was 10 years gap, it was revised. Now this recently it is already 4 years, it is getting revised. Yeah. Every 4 years it is getting revised. And recently is the 9th edition, is 2022. Yes. There are 4 volumes. Fourth volume is deals with the veterinary vaccines. And in this 4 veterinary, next slide please. I talk about the vaccines. There are around 50 vaccine monographs listed in the pharmacopoeia. Bacterial vaccines, viral vaccines, live and viral vaccines, inactivated. There are many. These are actually vaccines manufactured in the country, either by government or by private manufacturers. Imported vaccines are not listed here. Next slide. There are also so many antigens also listed in the pharmacopoeia. Next. Among the vaccines of the livestock, these are some of the livestock vaccines mentioned. That is for sheep and goat, there are 4 5 bulldog goats for sheep for PPR. Then canine, there are so many. Canine adeno, so many vaccines, canine adeno, parvo, corona, distemper, and infectious hepatitis and rabies in the bottom. And swine fever, for swine is only one classical swine fever, and cattle FMD. Although LSD is being produced in the country, it is not yet included, it will be included soon. Next slide. For all the monographs, structure is the same. It may be bacterial or viral, live or inactivated. Definition, production, identification, then moisture content. If it is a live virus, live vaccine, it is a virus content. If it is, there is only one difference between live and kill. If it is live vaccine, virus titer is the one standard, and if it is inactivated, serology will be the standard. After applying the vaccine, what will be the serological status? Then safety, sterility, potency, and storage and labeling. These are for all the vaccine almost same. Next slide, please. This is also for inactivated vaccine. There's only one difference in safety test. For live vaccines, you have to apply 10 times dose. For inactivated vaccine, only 2 times dose. This is the difference. Rest are the same. Next. Yes, already discussed 10 times and 2 times. Because we have to check by giving overdose, whether it is giving an uncovered effect or not. That's very important. Then sterility test we do into 2 different type of media. One is FGM and SCGM. This is already simple test. Yes. Next. Yeah, these are, so you can skip this. One more important thing is GPT, growth promotion test. Suppose you are doing sterility test and you have procured some chemicals from some company, and you are testing and you say that okay, it is passed, sterility passed because there is no growth. But how you are sure that this media is maybe a substandard media, it will not promote the growth of these contaminant, so you have to do the GPT test. GPT test, whatever on the left side, all these bacterial strains, ATCC number given, these strains are available, 80,000, there's a package, you can get from high media or other companies. So, any media you are using, FTM and STM I was referring to, you have to test whether these media support the growth of these organisms, standard organisms. Then only you can use that media. And one more thing, this is we talk about AMR, that antibiotic is antibiotic. Many companies or many state biologicals, they may do this practice, suppose to get the vaccine passed in sterility test, they may use large amount of antibiotics. So that when we come to regulatory authority or testing lab, we pass it no growth. But how do you know that antibiotic is added? This is the test. We add media, we add vaccine, and we add these organisms. If the organisms are not growing in presence of the vaccine, that means huge number of antibiotics are used. So that we can catch board. Next book, please. Efficacy by challenge study and by potency. Challenge study, is very tedious and painful to the animals. If a challenge study has been done in a good number of batches, we can the challenge study, and then we can go only by titer. There are so many vaccines are there, classical fever, then PPR, so many other vaccines are there where challenge study can be done away with. We can only test the efficacy by the titer. In under NHCP, we are doing PPR test, PPR and CSA by titer only. Because we have done enough challenge study, we have proven that it is correlated. Titer and challenge is correlating. For LSD, challenge is the only method now. So any batch we are getting, we are doing the challenge study for LSD.
So here that means if certain regularly using vaccine, you need not to go for a challenge study. You have to do challenge study only once you have done good number of batches. Some batches you do challenge study.
Once you have done some 5-6 batches challenge study, all are passing, then you can and you have a correlation study. Correlation study is the titer, alternative method. Then only you can do a use of challenge study.
Alternative strain, suppose CSF regularly using strain is okay. Another better strain now they develop. For that also challenge study.
Yeah, it's a new batch challenge study is required. Next. Okay, this is written where validated potency tests are available. Target species vaccination challenge study may not be required. Other tests is master content everything. Only one point I will discuss here. That is the residual inactivation life for FMD. Whether you have to check whether it is activated or not. That methods are available. Then interference test is there. Yeah, interference test maybe 6 point. It is important because for canine vaccines, there are so many combined vaccines, 6-in-1, 7-in-1 canine vaccines. 5 component of viral and 1 component of bacterial. What they do, they provide freeze-dried vaccines of freeze-dried cakes of the viral vaccines and liquid form of the bacterial vaccine. There you have to see that whether the liquid content of the bacterial vaccine should not affect the titer of the viral vaccine. That is why interference test. The bacterial vaccines or diluent should not interfere with any of the other vaccines. This is for combined vaccines. Interference test is a must. This is that I already discussed. Stability test. Many companies they write a stability test 24 months, 12 months, and they mention that they have done the accelerated stability. Accelerated means they expose the vaccine in high temperature, 45, 56. For 7 days, and they extrapolate the data. But many of them have seen that data is false. You have to do actual test. You produce a vaccine, keep it at recommended temperature, self temperature 2 to 8 degrees, and see till the end of the see for 6 months, 12 months, 24 months. If you claim that your vaccine is stable for 24 months, at 24 months, the vaccine should contain that therapeutic dose. What is required? For example, PPR. You claim PPR vaccine stable for 24 months. After 2 years, 24 months, the PPR vaccine should contain 2.5 TCR per dose. I've seen many companies, they overclaim. They have not tested for 24 months, but they just write 24 months. So that you have to take care. No, don't worry. Why you claim 24? If your vaccine is 6 months, why says at least it should be 6 months? It is good. If your vaccine is stable for 6 months, it is also good. Then there are actually in pharmacopoeia told 50 different monographs are there. Among these 50, majority are poultry vaccines. 40% are poultry, rest of less. So I separately poultry vaccines. Next slide. These are the different poultry vaccines. Viral vaccine, anicate. These are the 4-5 most common. RD, infects IBD, IB, Merex, and foulpox. Rest are produced but in less quantity. And among the bacterial vaccines, Coryza, foul cholera, salmonella vaccines are listed in the following. Next, please. Yeah, that type of vaccine inactivated live already discussed in the previous. The previous slide is for poultry. Actually, there are so many combination vaccines are coming: 2-in-1, 3-in-1, HBT vector vaccines containing different NDV and foulpox. This type of combinations are coming. And one more vaccines are now recently coming, like immunocomplex vaccines. It is coming for mostly for IBD. IBD, what happens after that hatching, the maternal antibody will be there. So in presence of maternal antibody, if it is vaccinated, it will not be protection. So if they are coming, many companies are coming up with this new type of vaccines. This is a combination of virus and antibody. Combined they will give. So when it is inoculated, that will not follow that normal immunology channel, immune production channel. They will bypass that, so that slow release of the antigen will be there in the system, and vaccine will come. Even in presence of maternal antibody. Okay, next. In poultry vaccines, in other vaccines, there are only cell culture or animal model. Poultry, you have option of egg model, egg also. You can inoculate the eggs, you can do the titration in eggs. There is no need to do cell culture on that. There are many vaccines protocols in eggs you can do, but provided it's better if you have SPFX. I don't know whether it's available here in this area. SPFX. You have to order. So it's not very costly also. You can do that in SPFX. Serum pathogen, specific pathogen free eggs. Next slide. For poultry vial, many poultry vials standards are already there. Some titer requirement is 3.5 EID 50 means egg infected dose. Or for Medex, you can see that in red color, 1000 PFU. Each dose should contain this much doses. Next slide. These are the method of doing HA. We all know. Is it titration? I will not spend much time. For IBD, for infectious bronchitis, you have to see that that curling, dwarfing, and stunting growth of the embryo. Left side, the top left side is the big one, is the control, and if we can know the titer based on this, and up to how much has dilution producing the disease. Next. Similar is for IBD also. This hemorrhagic lesions will be there in the embryo. Next. Many diseases, it is done in cell culture. We can see that class. There is a CP type class, but many diseases problem is that now in the country 2 bivalent vaccines are used. Could have serotype 1, 2, 3 like that. Serotype 2, 3. So to test that, you should have a monoclonal antibody against each serotype. First, you have to neutralize one serotype with the monoclonal antibody, and then do the titers. So double titration, and monoclonal antibody is the most. Next, please. This is a method of testing of the inactivated vaccines. Inactivated vaccines don't need to go for titration, only the serological standards. Some vaccine 160 for each titer, 120 like that. Some AGID 1 in 8 like that. Okay, next. Then regarding PPR, good pox, I have missed that classical science here. I'll just tell you. Next slide. Next slide. Next. Yeah. Here also we have to do that identity, sterility, mycoplasma safety, titration. External agent, there are methods, well-defined methods are there available, and potentially challenge method is required, and level claim. Level claim should contain how much dose, how much group, what is the stability, or storage temperature. Yes. For sterility test, there is a normal conception that 1 or 2 vials you can take and do the testing. No, actually for manufacturer you have to take 10% of the total batch size, or 10, whichever is less. So at least minimum 10 vials you have to test for sterility. For SCGM and FTM. Next. And virus titration as usual, there's a let down procedure are there and standards are there. The vaccine should meet that standard. Next. There are many viruses which produce CP. For those who have PPR good pox, you can see the CP. you can see that CP is minus 1, 2, 3, 4, up to 4 you get CP. You got CP and next minus 5 and minus 1 CP. That means it is 4.5 per, after the read statistical formula, it will come to 4.5 right? Then you have to match what is the dose size of the vial, so that it is having that therapeutic use. Next, this example of PPR also similarly. Next. Yeah, before this is a statistical method. Before that, I'm sorry, I don't have the slides for this CSF. CSF does not produce CP, so that we have to develop alternate method. So yeah, there are ideas we have developed a input FAT technique. Based on that, we have done some correlation studies also. 1000 TCID50 is equivalent to 100 PD50. In the pharmacopoeia, what standard is given only? It was given earlier. PD50, vaccine should contain 100 PD50. From 2018, we have included one more method that instead of PD50, we can do the high titration. Because we correlated 1000 TCID50 is equivalent to 100 PD50. So we can do high titration. And Dr. Bhavan sir, they are also doing the same. Next. Vaccine testing each manufacturing unit should have a vacuum moisture analyzer where you can measure the moisture as well as vacuum testing torch. It's very convenient. Easily you can see the result is done or before testing. Next. So we discuss test of external agents. There are many methods available. Like reconstruct the vaccine with, suppose for PPR, we have to reconstruct the vaccine with anti-PPR serum or monoclonal antibody, and then inoculate in the cells. If it does not produce CP, then no external agent is present. If it produces CP in spite of having monoclonal antibody, then some other viruses may be there. And another method is there are some hem-absorbing agents also. There are some viruses hem-absorb that chicken RBC. Chicken RBC, I missed that photo. Chicken RBC is an oval in shape. So if you infect the cells with some virus, some PPR virus, after 24 hours, you just put chicken RBC, raw chicken RBC, and incubate for 37 to 30 minutes. If any other agents are there, then this chicken RBC will absorb in the cells. Then instead of these clean cells, you will get some oval-shaped, oval-shaped structures. That means there are some stimulus agents available there. That is leading to the hand absorption. Absorption of the chicken RBC. So that will fail. Now the stimulus agent is present. That will just available. Next. This is the last. I promise this is the last slide. Potential testing of vaccines for LSD. If anybody is interested to produce in future, this is the challenging method. What we do, this is called climb titration. That's just like sick folks. We'll save the animals and different dilution will vaccinate. Will not vaccinate. One dose vaccinate in the next regime intramuscular. Now after 28 days, we'll challenge by this method, by climb titration. We'll do the titration. You can see top left one is the vaccinated and right one is the challenge control. If we put different dilution of the challenge virus, -1, 2, 3, 4, 5, 6, from left to right, you can say, there should not be any lesion production in the vaccine, but control should have some lesion. There may be some lesion in the challenge in the vaccinated also, but difference should be in the range of log 2.5. If the title is coming to 4.5, it should be less than 2.2, like that.
So one thing is that this particular potency testing, is it having a correlation with the neutralizing titer of serum in LSD?
Neutralizing titer we have not seen because many times you have seen that there is no antibody response even after vaccination or even after challenge.
That is what I am saying, sir, because in many cases, that is why they are saying that CMI, cell-mediated immunity, is much more important in LSD protection than humoral.
Yeah.
Because despite zero neutralizing antibody, also animal remains protected.
Initially, we were thinking that we will take help of this technology. We collected serum every weekly serum, even after challenge serum. We tested only 2 safety animals were positive for serum. None of the vaccinated animals had serum positive, but they were safe after challenge. Yeah. So that's why every batch we are doing challenges. It's very tedious. We have to go very tedious. But we are doing.
But this cell study, I think now we cannot PSL
facilities or like that.
It should be basal. Is it as per DBT is basal tree organism?
Animal BSL tree.
The Muktasar, you know, we have been there. It's a natural basal. We have a
Outcrop.
We have made all these insect-proof shades and very good shades and all. We have got all mechanism. But how the dung will be discarded, how the urine will be discarded, how to clean the premises, that we do, and we just check that movement of the virus from Well, this is the last. Next. Yeah, thanks for listening.
Thank you, sir. So I take this opportunity to thank Dr. Pranav, sir, Head Division of Biological Standardization, for a very enlightening and a knowledgeable presentation on quality control and testing of veterinary biologicals. Most often, I think government-run state biological laboratory, they produce vaccines, and even veterinarians, we should have an idea on how to vaccinate, what is quality of a vaccine. Just not enough for us to vaccinate it. It involves a very complex and tedious process. So this presentation has been, very enlightening and has educated all of us. Thank you again. So now I would request Dr. Anuj Chauhan, in charge of ITMU, ICR IVR Aizatnagar, to give us a brief idea on the available technologies of ICR IVRI from the animal health perspective. So Dr. Anuj, thank you.
Thank you, sir. Are the slides visible online? From IVRI?
Yes, it
somebody
is
can respond?
Yeah, visible.
Okay, thank you. So, good afternoon and a very warm welcome to the learned gathering. Honorable Director and Vice Chancellor IVRI, Dr. Trivedi Dutt sir, Director Research, AU Khanapara, Dr. Prabodh Bora sir, Joint Director Extension Education IVRI, Dr. Rupsi Tiwari ma'am, all the honorable dignitaries present in the hall and joined online. It is a matter of great pride and privilege for me to present the brief technology portfolio of IVRI, specifically in the animal health sector, which have been developed over years of research and innovation. Next, please. Yeah. So we at ITMU basically handle all the technologies which have been developed at IVRI. So we also are involved in the transfer of technologies to industry and other stakeholders, as well as the protection of IP in terms of filing of patents, grant of patents, designs, and copyrights. So Institute has been able to gather 32 patents for its different technologies, 22 designs have been registered, and 43 copyrights have been received for the technologies developed at IVRI. Alongside 44 technologies have been commercialized to more than 150 industries, entrepreneurs, government agencies, and NGOs, which have earned the Institute revenue of more than 12 crore rupees. Next, please. So the main forte or the main strength of IVRI is in development of vaccines and diagnostics for different diseases of livestock. Apart from that, we have strong presence in other sectors also, like feed additives, reproduction, value-added products, designs, and copyrights. But my presentation today will focus on the health aspect, that is the vaccines, diagnostics, and therapeutics. Next, please. So the vaccine technology portfolio of IVRI. So IVRI has developed several vaccines and which have protected the livestock population from various deadly disease. So as already the Honorable Director sir has told, under National Animal Disease Control Program, all the vaccines which are being used to control different livestock disease have been developed at IVRI, including the FMD, brucellosis, PPR, and CSF, as well as the soon to be marketed LSD vaccine. So the vaccine broadly we can classify into two, which have been developed: the conventional vaccines like for FMD, brucella, buffalo pox, and LSD, as well as the gene-deleted marker vaccine for brucella, delta per, and IBR. So next, please. Alongside, some new developments also have.
Right. You have to tell the vaccines for cattle, but each and each vaccine.
Okay. So to emphasize upon the different vaccines which have been developed, like like the Lumpy Provac, which has been developed as per the demand of the country following the outbreak of lumpy disease in India. So a live attenuated vaccine has been developed by IVRA in collaboration with the NRC, and this vaccine has been found to offer complete protection, and it has been developed using an indigenous strain. And this vaccine is conferring 1 year immunity on the animals which have been vaccinated. Then for brucella, there is one vaccine which is currently being used in the control program, cotton strain, and one more vaccine has been developed, which is the gene-related vaccine, which has been developed by deletion of pyrosamin synthetase gene, and this is denominated as S19 delta per, and it has DIVA capability. It can differentiate the vaccinated as well as the infected animals. So it has huge potential for use as marker vaccine. And advantage is same as of the previous S19 vaccine in terms of giving protection. Then for the IVR disease also, IVRI has developed one more gene marker vaccine, which has been developed by deletion of glycoprotein E gene from the genome sequence. And again, this vaccine also is a marker vaccine, which has capability to differentiate between infected and vaccinated animals. And this vaccine also has been developed from an indigenous strain. Thus, it has more scope of offering protection to IVR as compared to the exotic ones. And it will be hugely useful once we approach towards the status where we can declare the country IVR free. Then recent developments include the development of buffalo pox vaccine. So this vaccine is again a live attenuated vaccine, and this is also has been developed from an indigenous strain, the Vijayawada 96 strain, and it also offers a complete protection against the disease. Next, please. The new developments in the vaccine technology from IVRA include two new vaccines. Like the first vaccine is thermotolerant FMD. As we all know that there is a problem of cold chain breakage in the field. So many times complaint happens of vaccine failure. So IVRA in collaboration with DIFMD Bhubaneswar, we have developed a thermotolerant FMD O serotype vaccine strain using this strain, virus strain. And the advantage of this vaccine is that it is tolerant to a transient break in cold chain and can tolerate temperature even up to 45 degrees centigrade. And this retains all the vaccine worth attributes as similar to the conventional vaccine. And it has been found to be even a better immunogen as compared to parent virus in terms of neutralizing antibody response in both monovalent and trivalent forms. So this is in collaboration with DIFMD Bhubaneswar. So again, IVRA being a leader in vaccine, especially the FMD, we are going to soon launch a negative marker FMD vaccine, and it is being developed at the IVRA Bengaluru Center. And all the tests, like the vaccine potential of marker virus by immunogenicity studies, development of blocking ELISA, and immunogenicity evaluation have been completed. And this is the final stage. And very soon we are going to commercialize also this vaccine. Next, please. So our vaccines have been hugely popular in the field. Like when there was a national call for lumpy vaccine, we have developed it, and it has already been transferred to 3 commercial firms and 1 state biological at Pune. And this is in advanced stage of approval as the process told by Dharsar. So it has cleared many of those stages. And it is an advanced stage of approval, and very soon this vaccine will be in the market. For brucella abortus S19 delta per vaccine also, it has been commercialized to Hester Biosciences, Ahmedabad. And if you see in the bracket, these are licensed terms. So if anybody is interested in further buying these vaccines, for lumpy, the license fees is rupees 75 lakhs and 5% royalty on sales. And this license will be valid for a period of 10 years. Similarly, for brucella, these are the license terms. Next, please. Again, for small ruminants, as we all know, there are deadly disease, PPR, goat pox, and sheep pox, which have been plaguing our small ruminant sector for long. And IVRA has developed the solutions to tackle these diseases. And 3 very important vaccines have came from IVRA research: PPR, goat pox, and sheep pox. So PPR is being used. The IVRA developed vaccine has been used since last many years. And it is a live attenuated vaccine which gives lifelong immunity to the animals, and it is also being implemented under the control program. It has been developed from Suguri 96 strain, Indian isolate. Similarly, goat pox and sheep pox, they offer very long-term immunity to the entire life cycle of goat and sheep respectively. That is to the tone of 4 years, and both are of the category of live attenuated one and developed from the Indian isolates. Next, please. So if you see, it is a very popular vaccine from IVRI, and it has been commercialized to 8 houses, and including 2 biologicals. So this vaccine, if you this has generated a lot of revenue for us. So although revenue is not important, important is the problems of the livestock and livestock owners should be solved. But from the revenue, what we can see is that if we see the revenue amount and compare it with the present the vaccine cost, we estimate that around 65 crore doses of this PPR vaccine has already been used in the small ruminant population in India. So this is the kind of impact our this one single vaccine has created in the national small ruminant population. Next, please. Then again, one popular vaccine for small ruminants, goat pox, which has been again commercialized to multiple state biologicals as well as commercial houses. And this vaccine also has earned a good revenue to IVRI, which is again indicator of its popularity and impact in the field. And for this also, the estimate is that more than 35 crore doses have been produced in the field and used in the goods. Next, please. Then coming to sheep pox, again this vaccine has been commercialized to 5 commercial houses, and this including the Haryana Veterinary Vaccine Issue, the state biological. So this vaccine again is a hugely popular vaccine from the IVRA research. Next, please. So as per the mandate from ICR and Government of India, that we need to develop combined vaccine, the bivalent or multivalent form, so that the labor, cost and time is saved, and in a single dose we can vaccinate against the multiple disease. So IVRI recently has developed a PPR and goat pox combined vaccine. So it has the advantages of both vaccines. Both of these vaccines already individually were developed by IVRI, and they have been individually commercialized also. So now this combination vaccine has been developed, so it can combat both the disease from a single shot. Again, projecting a lifelong immunity to the vaccinated animals. Besides, it has huge export potential also. Here, I'd like to emphasize that similar to PPR and goat pox, we are also in the final stage of development of PPR and sheep pox combined vaccine. And very soon, that vaccine will also hit the market. And this vaccine already we are in process of commercialization. So very soon, the doses of this vaccine will be available in the market. Next, please. Then coming to pig, which is an important wheel of economic progress in the Northeast region. So for pigs, we all know the deadly disease is CSF, classical swine fever. So for classical swine fever, IVRI has developed a vaccine which is usually popular in field, including the cell culture vaccine, which was earlier there using exotic strain, and the classical swine fever vaccine using the indigenous strain. So this vaccine is again of live attenuated category, and it has been found to be safe and very potent in tackling the disease. It has very high titer. So from very less amount of cell culture flask, a large number of doses can be produced. And it offers more than 1.5 years of immunity to the vaccinated animals. And at 3 months of age, animals can be vaccinated. Again, this vaccine has advantage of giving better protection because it has been developed from an indigenous virus isolate. Then for swine septic, that is the pasteurellosis also, IVRI has developed one important vaccine, which is of the category of oil adjuvant inactivated vaccine. It imparts annual immunity, 1 year immunity against this disease. Then for a very important zoonotic disease, as we all know, Japanese encephalitis, the cases happens in humans also. There are a lot of infant mortalities, child mortalities also. So this vaccine is developed, although it is catering to the pigs, but indirectly it is benefiting the humans also, because it is breaking the infection or transmission cycle from pigs to humans. So all the areas where this Japanese encephalitis disease is more prevalent in pigs, So this vaccine has a huge potential. It is again a Vero cell-based inactivated JE vaccine developed from an Indian isolate. So it has better chances of giving immunity in the Indian conditions, as well as it imparts 6 months immunity to the injected animals. Next, please.
One, two, question regarding the CSF indigenous isolates production. From which particular indigenous isolates have been collected for production of the CSF vaccine? Either from the mainland or from the northeast?
No,
I
that
think
you was talking about previous vaccine. That is the Lapinase vaccine. Initially. Then what is the left side? That is isolated from MP from 1982. It is not from Northeast. But you know the CSF virus is only one serotype. One serotype belongs to 1.1 lineage. And the Lapinase vaccine also 1.1 lineage. That will give protection against all. I know why you are talking, because other strains 1.2, 2.1 are also there, but there are ample literature. There is 1.1 lineage will give protection across all these 2.1, 2.2, 1.2 lineages.
So basically, to prevent the handling of rabbits and slaughter of rabbits, we shifted from Lapinase to cell culture vaccine. Next, please. So again, this our vaccine has been popular in the field, and it has been commercialized to state biologicals as well as to commercial firms. And these are the types of royalties and license fees we have received. And these are the terms, because we are receiving interest from some companies and state biologicals, especially for the swine fever. So these are the terms for live attenuated indigenous CSF cell culture vaccine, which presently we are offering. Next, please. Then shifting to the poultry sector. So as Dhar sir was highlighting in his presentation regarding the infectious bursal disease and the problem of immunosuppression as well as the maternal antibodies. So IVRI has developed a subviral particle Gumboro vaccine. And this vaccine, so basically the vaccines which are present in the market presently, so they have a problem of causing immunosuppression and even mild clinical signs. And because of interference of maternal antibodies, they are not suitable for use in the hatcheries. So this vaccine offers the advantage in both those aspects, like there is no immunosuppression, as well as it is suitable for administration in hatcheries. And this vaccine already has been commercially tested also in collaboration with a private firm from Hyderabad, Globion. And they have tested in their stock, and it was found to cause no immunosuppression, intact bursa, fabricious, and all the good facets were seen in that trial. So this vaccine is available for commercialization, and we are hopeful. It is in process, and it will be soon commercialized also to one commercial house. This vaccine offers 6-month immunity, and these are the license terms. So again, it is very competitively priced, despite the huge amount of revenue which can be earned from poultry sector. Still, the price is very competitive, only rupees 35 lakhs for a duration of 15 years with 5% royalty. Then to another important species, the ducks. So presently, the vaccine which is available for this duck plague, it is imported vaccine, and it is costly because of all those import costs. And for that, we have developed a solution, a live attenuated vaccine using Indian strain. So this is when it has high titer of vaccine virus, it offers 6 months immunity, and it has huge scope, especially, of course, in the South India and the parts of other parts of country as well. Next, please. So besides the cattle, buffalo, sheep, goat, pigs, poultry, for other livestock species also, vaccines have been developed. Like for camels, we have developed camel pox vaccine, and this is again assigned to Agrinovate. For the dogs also, 2 important vaccine products have been developed for canine distemper virus and canine parvovirus. And again, they have been developed using the indigenous strain. So they offer better immunity or robust immune response. And they are safe at higher dose, and they are highly immunogenic. Next, please. So all these vaccines, as I told you, they are already have been commercialized to multiple firms. And these are some of the snapshots of the commercially produced our vaccines. under different brand names like Raksha PPR and all these brand names from different firms. Bio PPR. Next, please. So these are the snapshots of goat pox, CSF, and sheep pox vaccine being produced by different firms and available in the market. Next. So these were our vaccine, which is a very important portfolio. The second thing in presentation is for diagnostics. So this is again is our forte, our strength. So two important products I will highlight here for cattle and buffalo. One is the monoclonal antibody based blocking ELISA for FMD. So this is a recently developed product from IVRI, which has been assigned to Agrinovate some few months back only, and this is available for commercialization now. So this is a modification over earlier 3ABC ELISA, also developed by us, and it has used for all FMD susceptible species, including the wild animals. And the presently NSB antibody detection ELISA, which is used in the control program, this has two specific advantages over that. That is, there is no need of using the species specific conjugates and no need to standardize again for these things. It has very high diagnostic specificity, more than 99%, and more than 95% diagnostic sensitivity also is there. It has been validated at the Prime Lab, WRL Netherlands, across all the 7 serotypes for different species. So this is available for a license fees of rupees 10 lakhs from us. Then again for an important rotavirus which causes diarrhea and problems in livestock, synthetic peptide based antigen capture ELISA. So this has been developed as a replacement of imported kits which are being used presently in the country. It has already been assigned to Agrinovate, and these are licensed terms. Apart from the thing that it is cost effective, the another advantage is it is highly specific. There is no cross reactivity with other enteric viruses, and because this is synthetic peptide based, so there is no issue of handling live virus. So no biosafety issue also are there with this. Next, please. Then for sheep and goat also, numerous diagnostic technologies have been developed from IVRI, including for the blue tongue disease, like blue tongue sandwich ELISA, competitive ELISA. The name itself signifies what is the technicalities involved in these, and they have been assigned. So also against the PPR. And these are the snapshot of those kits. Next, please. So these diagnostic technologies, they have also been commercialized from IVRI. Although it is difficult sometimes to commercialize the diagnostics, but our diagnostics are also being used in multiple places, like this 3ABC antibody NSP. This test is being commercialized, and this firm is producing OBU Biotechnology Services from Kerala. Then Vivimed also has taken from us this recombinant antigen based indirect ELISA, as well as Intervet has taken PPR hybridoma clone from us. Next, please. Now again shifting to pigs, because this is a huge big sector here in Northeast. So for the zoonotic disease of Japanese encephalitis, two important kits have been developed from IVRI. So one, the kit which is mentioned at serial number 1, it is for sero monitoring of the pig population. So this is IgG ELISA. So this is used for sero monitoring. Again, it is cost effective as compared to the imported kits which are presently being used, and it has other advantage, that is recombinant antigen based kit. So again, no biosafety issue here also. So this kit has already been commercialized to Messrs. APS Life Tech Pune, and second kit also is there for Japanese encephalitis, again IgM ELISA. So this like the first kit was for sero monitoring of infection. This is for checking for active infection in the pig population. So both of these are priced similarly at 2.5 lakhs and license has been given for 10 years. Next, please. Then to another important species of poultry. So for here also again we have developed 3 important diagnostics. The 3 are listed here for against infectious bursal disease, infectious bronchitis, as well as Newcastle disease. So all these 3 kits have been commercialized to Messrs. Carl Genomics. I would like to present some advantages of these kits over the already existing kits. So number 1, as with all Indian kits, they are cost effective. Number 2, that they use less amount of reagent, because in a single microtiter plate, around 48 samples can be tested. While the present serial dilution ELISA, hardly 8 to 10 samples can be tested. Then these kits are specific to all pathotypes of viruses. Again, these kits are using the recombinant antigens, so there is no biosafety issue in terms of handling of live virus. So these four basic advantages are there for these kits, besides, of course, a very competitive price. Next, please. So again, this is a new technology which is soon going to be coming from IVRI. So PCR-assay for species level differentiation of Mycobacterium tuberculosis. It has very high diagnostic sensitivity, specificity, and accuracy. And another advantage is that it can be used in both animals as well as humans. And if you see, presently no such single step multiplex PCR is available. So very soon we are going to complete all the proceedings for this technology also. Next, please. Now, after the vaccines and diagnostics, the third important aspect of the health we have is therapeutics. So we have developed some very important products. Like number one is our eco-friendly herbal acaricide. So the tick is a huge problem in India, and the presently used chemicals, the synthetic pyrethroids, the organophosphates. So many ticks, they are now developing resistance. So the farmers are spraying all types of insecticides, but ticks are not dying. So for that, we have developed a product which is herbaceous in nature, and all these plants are widely available across the country. And we have tested in the different dairy farms in Punjab, Haryana, as well as, of course, in our own state of Uttar Pradesh. And it has been seen where the chemicals were not working in these ticks. Our acaricide has shown 80 to 90% even against these chemical resistant ticks. So this also additionally has fire repellent property, and this passes again all the OECD parameters for use of safety in animal application. So because this is herbaceous in nature, so the biosafety issues also is normally tackled as in all food products. It has been commercialized to Ajay Biotech at the cost of rupees 20 lakhs and is further available for commercialization. Next, please. Now, two important, again, one more is acaricide and one is the problem in field of diarrhea. So although antibiotics are available, but there is problem of AMR and issue of the residual toxicity in feed. So we have developed a herbal product which is tackling the condition of calf diarrhea, and within 3 days this herbal product has been shown to have marked improvement in dehydration and depression. So this is cheaper than equivalently available herbal formulation, and this is the doses which is mentioned here. If you use this for 3 days, they will effectively treat the diarrhea condition. And it has also been recently assigned few months back to Agrinovate and is available for commercialization at these terms. One more herbal acaricide has been developed from IVRI. So this again has good effect against the tick population, even larva, nymph, adult, and it is again safe and easy to prepare and has same advantage as previous one. It is eco-friendly and environmental because it uses the herbaceous plants. Next, please. So two more therapeutics I would like to emphasize here. One is the Masticure for formulation to prevent and treat mastitis in bovines, and one advanced technology developed using the modern biotechnology tools and the stem cell technology. That is a mesenchymal stem cell based formulation. So important advantage here is the wound healing rate is faster, especially in the prize dogs. So many dog owners are willing to spend the money, especially the prize dogs, and it leaves a minimum scar. So if you use the normal any antimicrobial products or any creams, scar formation will be there. So it reduces the scar formation, and even better than the normal stem cell because it is based on conditioned media. So it is cheaper than stem cell, cost effective than even stem cell. Next, please. Because we have the animal husbandry directors here. So apart from the health, I'd like to focus one more technology of ours, from waste to wealth, which is very farmer friendly. And a new, this is a jag, which has been named as Jag Opal, which is better than the earthworms which are existing in the country. And why it is better? Because it has much higher fecundity. and it has more heat tolerance. Like in Indian conditions, we have temperatures to 45 to 48 degrees centigrade. So this lives better in that temperature and has high fecundity, and it is a hugely popular technology which has been commercialized to 64 entrepreneurs, NGOs, farmers, as well as government issues. Next, please. So from the health aspect, numerous technologies have been transferred, and these are the kinds of revenue which have been generated from the sale of our technologies, which again are indicators of the successful use of our technologies in the field. Next. And this is the nationwide recognition which we receive often for our different products, like for the lumpy, which we have taken the challenge and produced a vaccine, which was released by Honorable AM. Next, please. And these are the release of different technologies which have been developed by IVRIs, by the high dignitaries at ministries and ICR. Next, please. And our technologies have received wide news coverage, and these are some of the snapshots of the coverage of our technologies. Next, please. And as a indicator of our success, we have been rewarded. So one slide back. Like this recent reward we released for development of lumpy skin disease vaccine, and this award was received during the ICR Foundation Day. So next, please. So this was all regarding the ICR IVRI technologies for health aspect. And thank you for giving me this opportunity.
Excuse me, sir. We have been discussing with so many vaccines and all, no? But the Northeast is now suffering from this ERS, African swine fever. So many farmers, they started saying that we are not willing to rear pigs also, because once the pig grows, it becomes big and they'll be attacked with ASF and they will die, and we are helpless. We cannot do anything. We simply tell them that yes, we have to do biosecurity, this and that. That's the all thing. And they are asking for vaccine with us. So we cannot say anything. And we want to know what is the thing means, what are the problems in manufacturing this air.
Yes, sir. Your concern is all of our concern also. African swine fever is the history, it is 100-year-old disease. Plum Island and the Fulbright Institute, they are trying in long time this vaccine. But it's not, only 3 vaccines are available in the world now. It is being released from, official release from Vietnam. And those vaccines have been produced with collaboration with Plum Island. Okay, and that is based on Jordan strain. They are also type genotype 2. India also having genotype 2. But there is ample literature that this vaccine will not be for protection to heterologous strain. Suppose if we use the Jordan strain, we can import from the Vietnam, and they may not protect in India against Indian strain, because all the vaccines have been released officially. But if you go Google search, if you go into deep into that, none of the vaccines have been tested by challenge method. What method they have tested? They have manipulated, they have seen the serum, and they test only serology. But we don't know yet whether those 3 vaccines will give protection against India or here Dr. Burman is there. He has because this African swine fever is a BSL-3 agent. We cannot handle everywhere. We have very limited facilities, like Assam Medical University, they have a BSL facility. Here they are at IVRI, I don't know, we have a BSL facility. Some scientists are already started working on that, yes, sir. And Dr. Burman is also working, but it is long way to go before we tell that we have developed a vaccine. So, and government of India also, that these companies, one or two Indian companies, manufacturing companies, they have approached government of India to import some of these vaccines from Vietnam. It is under discussion, and we recommended a good vaccine is required from IBA contract. We recommended that good vaccine against ASF is required, but its efficacy should be tested, and all this should be documented. We should have that paper that is, we should ensure that we give this protection against our Indian state. Then only we can that's
in fact, in this regard, can you just give us a rough idea of how much time is it going to take if we have to get it from the vaccine.
Once that, there is a expert group under the DASD, under the Animal Commissioner.
Empowered group.
Empowered group. If they approve it, then it will be imported immediately, 2-3 months.
Let me supplement Dr. Dhar. In India, already under NASF, the project has been given to Nisar Bhopal, wherein they are also trying to develop an indigenous vaccine. So I was a part of NASF during the review. So that is one, and also Dr. Nagan Burman's lab, along with the teachers from Animal Pathology division of this institute, also have tried to develop a protein cocktail vaccine against the ASF. So this is only initial stage. You cannot say that it is going to come to the commercial stage in the near future. But these are some beginning. As already he has explained that for almost 100 years, people have tried to develop a vaccine, but nobody has become successful. Even in Vietnam, if you see the literature, after it was introduced to the field, it was again withdrawn by the government itself because there are so many complaints of unexpected sort of interactions and some, so to say, adverse effects. So that's why the people have started complaining and government withdrew those vaccines in Vietnam, in the original country itself. So we should not jump into the conclusion that the vaccine will be available in India and that will be very effective to protect Vietnam. We should not be in a hurry to procure that from Vietnam because in the producing country itself, there is a lot of controversy going on regarding the vaccines available in Vietnam. Thank you.
Even though if we are government of India have given the permission to import, after that the vaccine has to be undergone in various regulatory tests also. Then only finally can be said that it can be used safely. So I think even though imported vaccine it is a long way to tell.
Here, sir, some alternative measure we should try to develop or formulate because vaccine definitely will be challenging to get immediately. Alternatively, our pig farmers or all directors are here. We should try to go back to a very organized scientific learning system because Corona teaching us a lot of things. We learned hygiene, biosafety, compartmentalization. So we should work out on that line. Some the protocol we are developing about the compartmentalization in the organized farm and semi-organized farm. So with this compartmentalization, safe and biosecurity, to some extent, I think we can save the farmers. In fact, with our suggestion, I got the result. 4 or 5 farmers they are maintaining safely. So these things, if we can propagate and encourage,
This package of practices, I think this should be popularized immediately. Because you see, this is the only way out at this moment.
So this guideline is already published by NRCP and it is an ICR website. How much percentage of sodium hypochlorite? 2%. How many days? For 30 minutes.
Biosecurity, proper biosecurity and biosafety measures.
All in details they have.
In that, IDRA has also taken initiative. We have developed a beautiful application, mobile application. That is, I think, totally for this what should be the biosecurity and biosafety measures for all categories of livestock. So I think this, at this moment, I think this is the only way we must popularize, we must handhold our farmers using this practice.
Another issue actually compartmentalization what I am emphasizing because this virus is low contagious,
yes, sir.
Highly virulent. Even the in contact say that those animals we can save. If we found the infected unit, clean it totally, clear it, next unit we can save. So that compartmentalization concept if we can infuse along with the biosafety. So that thing I think we have to train them and explain the principle. Not only all these website, all these things, practically we should work with them. Then they will realize and they will get the benefits.
Now, point comes again, the capacity building up.
Specifically, I have two queries to Dr. Chauhan on his presentation. The first one is that you have always shown that so many technologies have been developed as well as vaccines. We are all aware of these developments taking place in the last more than 100 years in IVRI. And all the stakeholders are really grateful to scientists working in IVRI for producing vaccines that are required for different conditions to protect the animals. So in the last several years, most of the Institute of Veterinary Biologicals working in different states are dependent upon the seed from IVR for producing different vaccines. That is well known. But now in the list that you have provided, we have seen that several new vaccines have come up, so which have been either commercialized or ready for commercialization. Now, the first question will be that whether all the technologies transferred to different commercial farms are being commercially produced now, so that they are available for the farmers to procure. Because many times that happens, we transfer the technology to the commercial farms, but they fail to come up with the commercial sort of brand in the market so that it is available in all the places in the country for the farmers. That is number one. Second point is that you have already given a price tag to each of the technologies and the vaccines that you have developed. So I think for commercial transfer to certain business agencies, these price tags are essential. That is okay, that we all agree to that. But is there any provision within IVRI to have a separate price tag or a negotiation to the Institute of Biologicals in different states of the country, particularly northeastern states, because we may not have the capacity to afford to provide that much amount of money to IVRI for transfer of technology. If it is available in a cheaper cost, or maybe it is also handheld later on by providing training, capacity building, and continuous, so to say, handholding during production, at least in the initial stages. That kind of arrangement or negotiation, if we can make with IVRI, I think it will be better for the northeastern states directorates to procure those seeds and to start producing a lot of other vaccines which are not produced by the Institute of Veterinary Biologicals. I know Assam Institute of Veterinary Biologicals producing 5 or 6 vaccines, and they have been produced since last several years, maybe more than 50, 60 years. They are not including any new vaccine into the fold because the technologies are not available to them at a cheaper cost. I think I would like to know from you whether such arrangements can be made.
Answer one by one. First is they have to let any vaccine technology is given to the companies. It has seen the light of the light was there. Yes, I am going to give example of two vaccines have shown, PPR and CSA. PPR vaccines you have transferred technology to 5-6 companies, even state governments, and now this vaccine is now being used in the national control program. Whatever we are seeing, this is they are using. CSA also we have given to a company, IAS. That is the first vaccine coming in the market. Any farmer, any from Northeast, I got many phone calls. So what you can procure? I just give the name of the company, they procure. In Assam also they procure. This is also used in the. There are some other examples. Now regarding the price, for example, certain vaccines, CSA or LSD, it contains that IPR. It is 75 lakhs. It's not huge, but it is a but suppose you give free of cost, low cost. What the companies may do, this can pass to other channel to them. They will also start producing. So it is very difficult. We can revise the rate to some extent, but not free of cost or not very cheap.
We can make a separate price range for the
government
and the
private.
that
That
will empower the state.
Only 40 years they can while they are going back, we can think of.
Yes.
This is Dr. Pramod, he has a very valid point. That for the state biological in the northeast state, because you see, because although we have complied, but Government of India have a special package for northeast. So we should also have a special prices for northeast. So this I think we must do. Thank you. Thank you.
So, sir, now I would request the next speaker, Dr. N.S. Burman and even Dr. Prabodh Bora, sir, to kindly participate and make remarks on the status of veterinary biologicals in Northeast region and also on the extent of emergence of these transmitted diseases and their control.
And also the capacity building.
And capacity building of the veterinary personnel belonging to different stakeholders. So please.
Thank you, Dr. Sen. Honorable Director, sir, Madam Tiwari, Director of Research, Dr. Bora, Arnav Sen, and colleagues from IVRI Kolkata Regional Center, and all the state directorates of the Northeastern region. And really, this is an unique opportunity for all of us that Mecca veterinary education, they are coming to the Northeast. And we always have the strong belief that whatever I are producing, this will be unique and quality things. And that same way, they should also share all the technologies with all these state countries, different region. So with that hope, veterinary biological also, next slide, started long back in 1950. It was in the Senikuti, and thereafter it is shifting to current location in 1966. And looking into the situation in Northeastern region, this is really a biodiversity hub of the country, and region is also very vulnerable for the emerging of new pathogen, human, animal both. And if you look to the history, that Rinder test, as history says, entered through Northeastern region. Chinese Khan from Mongolia, who aggressed invade the India, along with the bull card, he carried also the Rinder test virus, and they are responsible for spreading to the Europe and other countries. Secondly, the avian influenza Northeast also recorded in 2007 and 8 in Manipur and Assam. And like that, ERS first time recorded in country 2013 in Mizoram. And ASF recently, you can see along with the corona pandemic 2020 and LSD 2020 entering through Bangladesh. So Northeastern region, and as directors are actually said, 4,500 kilometers, the international boundaries are totally porous. If you look into the Manipur, that is the region freely people, product, animal can move from one end to another in the Sampai district of Mizoram. So like that, many regions are there which are really unrestricted, and as a result, all the emerging viruses in animal, also in human, enter through Northeast. And if you do not address Northeast, your mainland will not be safe. Although it is connected to a small 22 kilometer chicken neck, but it is proving once it is entered that African swine fever, it is spreading to the entire country. LSD entire country, avian flu. So we must focus, as the government of India, they rightly they emphasize that act is policy. So I therefore really request our director, sir, he should give this. They are helping because we have done a lot of collaborative work with the IVRI, and many graduates are graduated from the IVRI. So that way, the manpower we got, trained manpower, but at the same time, technology and other facilities, we should go for hand in hand, and money should not be, you can earn money from other sources, but not for at the cost of preventive measures, because this is the only right technique by which we can save the farmers. Our target is the farmer. Our target is not that the institute, how much revenue we are generating. That should not be the competition, and we should focus on the farmers, because poor farmer, ASF, everybody is crying because director are answerable, sir. That's why they are very much concerned. So looking into that, I'll see the next slide. That IVRI, that our Institute of Veterinary Biological established in 1950, when country was suffering from the So goat tissue adapted vaccine was developed in the Muktesar under initiative of Allen, Dan, Lingard. I think they were also the pioneer of people there, and subsequently transferred to the all biologicals and instead of veterinary biological Khanapur, Guwahati, which was located in Senikoti, they started with the goat tissue adapted rinderpest vaccine. Because of this process initiated, I think country claim we are free from the rinderpest. We must salute to the veterinary professional workers, right from the field assistant to the doctors, how much they have done. So this is the contribution of Institute of Veterinary Biological. Second disease is the BCPP, which was only prevalent in the Goa, that is Dhemaji, Lakhimpur and Tinsukia in certain pockets. Because of the constant production by the Institute of Veterinary Biologicals and supplying in the right time this vaccine, now country claim free from BCPP. So during early period when there was no commercial vaccine supplier, these biologicals providing lot of support. Even the rabies, when there was no rabies vaccine, this is the veterinary biological, they developed the rabies vaccine. Next slide. See the requirement for the entire northeastern region. If we consider the 80% livestock should be vaccinated, in that case we have a huge demand in pig for a single vaccine. If we want to give 2 times in a year, then around 68 lakhs doses we require annually. Cattle vaccine 212 lakhs 2 times if I consider. Then goat vaccine also around 86 lakhs doses. Buffalo 8 lakhs and sheep 6 lakhs doses. These are the huge requirement. So to meet up these things, next slide. Definitely we should strengthen our veterinary biological. The infrastructure wise they have sufficient. We have to improve it and modify it. They can definitely cater the need of northeastern region. And right now the bacterial vaccine they are handling anthrax, HS, BQ, enterotoxemia, and then viral vaccine, CSF, although in the Lepinized vaccine, but cell culture vaccine slowly they will adapt it. Ranicate disease vaccine, RD vaccine, F1 and R2B, dark plague vaccine, it is CM, chorioanthric membrane, the root production of vaccine, and then fowlpox vaccine. These are existing vaccine, all are procured from the IVRI. And institute, Assam Agriculture Institute, looking into the need of the northeastern region, we are developing some local candidate. So definitely in that case, we cannot go further without taking the help of the IVRI or other leading institute. So there we need your support so that whatever the candidate we are developing, these are potential candidates. So if we can improve and complete all the validation process, definitely the region will be benefited. Next slide. And presently, as because many commercial vaccine producing companies are coming up, so there will be limited demand from the dispensaries. This is the requirement for the state of Assam. So they are producing hemorrhagic septicemia currently as per demand 2.5 lakhs, black quarter 1.5, anthrax 1 lakh, then ranicate 10 lakhs, R2B 10 lakhs, and dark plague 15 lakhs, swine fever 5 lakhs, so this is lepinide 1, and fowlpox 10 lakhs. So they definitely, along with the advancement of technology, they will switch over to the cell culture technique, what IVRI developing as well as we are also developing. Next slide. So these are the status for IV, the Institute of Veterinary Biologicals, looking into the need of the northeastern region. The Assam Agriculture University, the microbiology, the animal biotechnology division, together we are working for development of the cell culture vaccine. CSF lepinase vaccine actually procured from the IVRI, that was we adapted in the PK15 cell line. In fact, the Thor will agree that the cell line I provided he is using. Or sir? Yes, sir. But the PK15 cell line I provided him. So he is maintaining. I am really thankful to him. So that like that DPV vaccine, which was initially in the CM, that vaccine we switch over to second embryo fibroblast, and we are developing one local dark plague virus, the KM19. This vaccine we have done adapted in the chicken embryo fibroblast simultaneously in the cell line, Vero and DF cell line. So this vaccine, the uniqueness what we have seen in the face of outbreak, that is as the emergency vaccine it is working, sir. I tried in the different field level. So when outbreak is going on, in contact bird we vaccinated in this vaccine. Within 5 days, the spread of the virus spread is stopped, and we analyze the mechanism. The IFN gamma is producing within 4 days of the vaccination. So this IFN gamma giving early protection, and subsequently by 14 days that humoral immunity is coming up, total bird flock will be protected. So farmers now they are approaching for vaccine that when there is outbreak they want vaccine. Means even outbreak when they are giving, majority bird would be saved by this emergency vaccination. So other goat pox vaccine, this is the local strain we isolated, and it is immunization trial already done. And this goat pox vaccine also we tried in the cattle in the higher dose. In the goat, we have given 10 to the power 3. Now in cattle we have given 10 to the power 4. It is giving a good CMI response as well as humoral response. First initial clinical trial we have done in the cattle. So like that, all vaccine after this climate change, I have seen the pox groups are coming up big way, and all producing a very severe lesions. Lot of the complaints are there. The mortality in the kid, but adult will lose the weight because of not taking the food properly. So all vaccine also we adapted in the varicella line. Now in the duck we have seen most of the duckling suffer from the pox. This is the new, I think 4-5 years we have observed majority ducks suffer from the pox. So that virus we isolated, and that pox virus also adapted in the varicella line. Like that, African swine fever, as said rightly, because it is struggling the all developing countries. Still then, when there was an outbreak 2020, immediately we submitted a proposal in the DBT, and they provided 2 years fund. With that, we developed good diagnostics and some protein targeting to 4 genes, and now we are planning to add 3-4 genes. This protein cocktail mixer, we have done one phase of immunization trial in the pig. It is giving a very encouraging humoral and cell-mediated immune response. But question is that whether it will really protect or not. So for that we need the NISAP help. So in that case, actually the National Institute, your help is required to facilitate that things, so that they will provide us this access for the saline study, whether it is working or not. Otherwise, we have to modify the technique. And many times we will approach the National Institute to give some other technological support. So it is not that National Institute will collaborate only with the National Institute. Some institutes are really potential. We have the problem faced issues. We are collecting, we are addressing. But if you support us, I think country will lead in a big way. So these are in the CSF. Thorsar said, although 1.1 giving the cross protection, but now 2.2 is coming up, replacing 1.1. Recent outbreak, Thorsar will also agree. So majority cases 2.2 is taking over the 1.1. So we should be ready with the 2.2. So we have some isolates from the Tripura 2.2 that we are adapting in the PK15. So we should have the readiness. And in that case, with IVRI, I did one collaborative project with this Praveen Gupta, the Diva CSF vaccine. So that was completed nicely. Like that, presently on CIRCO with this Madam Sohini Madan. The trial immunization trial, we are doing these things in the peak. So this type of collaboration, even with the Sai Kumar, the national network project on CSF, when Arnab was also there. So that type of collaboration we are really getting, and we want more collaboration. And Rimerella, another along with the duckling, sir, Rimerella, it is coming up in a big way. Many duckling will die suddenly, showing all the nervine symptoms, confusing with the duck plague, but that is not the duck plague, that is Rimerella. So that isolate also we have isolate repository characterizing, and one inactivated vaccine we have tried in the bird, it is giving good result. Next slide. So regarding certain technology, one DP daplet seed we transfer to Birak, our student startup. This program we shared with that one local DPP seed. Another technology we transfer to the thermostable NDB. Pankaj Deka, he is here. So he developed that thermostable NDB. Now Hanky Group, they take over this technology in, I think, a very nominal price, but that is giving a good result. So what regional institute we are developing, these are also very useful because they are appreciating. The isolate is giving a very good response in their different trials. And another diagnostic what we are developing in the African swine fever, because we do not have any indirect ELISA, sandwich ELISA. This ELISA will cost 1.5 lakhs. The foreign ELISA, that kit if we import. So now this P30 protein based ELISA, it is giving a comparable result. So that things, if we support us, we can validate and transfer the technology to the private companies, and multiplex PCR and another PSR, polymerized spiral reaction, because kit we should develop targeting to our the end user. So point of care diagnostics should be the priority, so that early diagnosis will give the opportunity to take the early decision. So that is why that molecular based PSR, polymerized spiral reaction, that is working very with the encouraging result. Within 10-15 minutes we will get the even PCR based result. So this type of technology we are developing, and I repeating the same thing, we need your support, sir. Next slide. All the diagnostic whatever we have developed so far, E2 based classical swine fever, indirect ELISA, then African swine fever, sandwich ELISA, antigen based and multiplex PCR. In pigs we have seen not only the single pathogen entering, there are the co-infection. During African swine fever, SIRCO was the invariably co-infected virus we noticed. So one detection of one virus will not give the conclusive diagnosis. We need a multiplex based diagnosis so that we can eliminate all probable viruses and get the real direction. So these are the diagnostics we are trying. Next slide. So this region, as I told you, whatever the isolates, tissue, serum of the all different categories, livestock, wild, or other the animals, we are trying to maintain. But maintaining of repository is really challenging. If there is no constant financial support, all these things, it is very difficult. But this is the precious things. We maintain isolates, tissues, and serum. Many companies now are they are approaching for validation of their kit with our serum. So these are the opportunities we have seen plenty, but your guidance, your support is very much necessary. Next. Yeah, thank you, sir.
Thank you, sir. Now, Dr. Prabodh, if you have some points, please.
Thanks to Dr. Barman for highlighting the initiatives taken at the level of the Assam University, Department of Veterinary Science, particularly the Department of Microbiology and the Department of Animal Biotechnology of the faculty are involved in all these efforts to develop certain locally produced diagnostics as well as vaccine candidates. I think already he has explained a lot regarding the necessity of having further collaboration with national institutes like IBRI and ISHA for further validation and standardization of the products that have been already initiated at our level. So as the opportunity for some of the advanced level validation and diagnostic development is not available with the state university. So I think it is always pertinent to have the collaboration with national institutes like IBRI and ISHA. Particularly in case of emerging diseases like ASF or lumpy skin disease or for that matter avian influenza and others. So always it is necessary to have collaboration for testing and validation of the diagnostics as well as vaccine, which are initiated at the state university level with NISA. So in that case, already we have approached the director NISA as well as some of the scientists, both individually as well as at the institutional level. And only last week I talked to Dr. Rajiv Kumar at the initiative of the chairman of the Agricultural Commission of Assam. I think you know Dr. S.S. Gupta. He was also for a long time the chairman of the NASF review committee. I was also a member of the committee for the last 6 years. So we had very good, so to say, rapport with the committee members. So with his initiative, we have tried to develop some sort of collaboration with Dr. Raju Kumar, because already Raju Kumar has been given the responsibility to develop the ASF vaccine from NASF. So can we have that kind of collaboration with IBRI in case of the validation and further, so to say, development of certain candidates to the commercial vaccine level? Because the testing and trial, whatever is necessary for final commercialization of a vaccine, may not be available to the state universities like Assam State University. And a collaboration with an institute like IBRI will be very much helpful for the teams which are working on that. So we look forward for your hand holding, the guidance as well as collaboration, so that these candidate vaccines can see the light of the day at one time. Of course, we are very glad and we feel proud to say that some of the vaccines have been already transferred either to the commercial farm or to the local institute of veterinary biologicals. And some of them are in pipeline. So particularly, I would like to demonstrate pride of introducing Dr. Pankaj Deka, who is an assistant professor in the Department of Microbiology. At one time, he was our student, now our colleague. He has recently transferred the thermostable anti-vaccine to Venkies. And after several rounds of trial, they have informed us that the vaccine has been found to be very promising and they are getting very good results in the trials they have initiated. So we are hopeful that in next 6 months to 1 year time that will come to the market. And they are planning to commercialize it in around 33 countries of the world. Because this is a unique kind of vaccine that does not need cold chain management. And particularly developing countries like India and other third world countries, it will be a very useful asset because we cannot maintain the cold chain. That is why there is a loss of potential of the vaccines many times. So I think that kind of work can be pursued in agricultural universities further with little support and guidance and the collaboration needed from national level institutes. I think in this meeting, I think it is a very good opportunity for all of us to discuss about that and how best we can go for having further collaboration in research. And at the same time, I would like to request you, because already we have an MOU signed with IBRI, that's a big opportunity for us. And the first PhD scholar who did PhD under my guidance in this university, but got the degree from IBRI. I think you know, under that MOU, Dr. Lukumani Uragunai, he was registered to IBRI because he, after completion of coursework, he joined my department as assistant professor. I was professor head of the department at point of time in biotechnology. So with the approval of the MOU from both the parties, we approached the then director of IBRI so that he can pursue his research under my guidance here and get the degree from there. So that was materialized. So that kind of, I think, sandwich type of research at both masters and PhD level can also be encouraged, so that to get the solution to the local problems in Northeast, our students can carry forward some advanced research with facilities of undertaking some part of the research work in IBRI national laboratories. So that kind of collaboration will be really helpful to find solutions to the local problems of the Northeast, as well as the students may be also trained well so that their capacity building activities will lead to further development in future with the manpower recruited in the different veterinary departments in the entire northeastern states. So I think that kind of collaboration, if we have, if we extend it further, some students of IBRI can also come and stay in our laboratories for 6 months to 1 year and conduct certain research under the able guidance of selected efficient guides, local guides, co-guides in the Assam University, so that we can also find further collaboration in research and get solutions to the prevailing problems in the Northeast. So that is my suggestion. Thank you very much.
Thank you, sir. So I think that we have come to the end of session 1. So I request Dr. Bablu Kumar for the next.
Thank you, sir. Now we are moving to technical session 2, which will be chaired by Dr. Ruchi Tiwari madam, and it will be coordinated by Dr. Rohit Kumar and Dr. Anuj and Dr. Suman Kumar will act as a reporting. Now I request Chairman Dr. Ruchi Tiwari ma'am to take the proceeding further. Thank you.
Thank you, Dr. Kablu. Thank you to Dr. Arnab Singh for having a very nice technical session one. There are a lot of inputs from our presenters as well as from our dignitaries from the Assam Agriculture University. Now we are proceeding to technical session 2, which will be co-chaired with me by Dr. Bablu Kumar and rapporteurs Dr. Anuj and Dr. Suman Kumar. So now we start with the remarks of the Director of State Animal Husbandry Departments of the Northeastern region. So I think if we have some representatives from Assam here, State Animal Husbandry Department Assam representative. Your remarks, inputs or constraints, anything that you would like to deliver here.
We are producing vaccines in the Institute of Veterinary, and research is going on to produce anti-toxemia vaccine also in collaboration with College of Veterinary Science. Dr. Naveen Mohan sir, he was my co-guide and he helped a lot during this process.
Please speak loudly, you are not audible.
We are studying on production of anthrax vaccine also, and we have started production yesterday only. We have prepared a batch of anthrax vaccine. So I want to thank Dr. Mohan sir for his help and entire guidance in this process. So we are producing vaccines in the IVD, but there are many constraints. We are planning to develop GMP and GMP. So the process is under consideration for GMP appliances and all. And we are trying to set up a cell culture laboratory in our biology section so that we can develop various cell culture vaccines in IVV, Khanapara. So I want to seek some guidance from IVRI in this regard. And for establishment of GMP in IVV also, I want to have some knowledge and guidance from her also. And there are constraints. I can say that our manpowers are not fully trained in the advanced techniques for development of vaccine. So if you can provide us hands-on training on production of vaccines in IVRI, then we will be very much grateful to you.
Thank you, ma'am. We have noted down your issues, and it would be better, can we have one letter from your department to IVRI regarding the handholding for establishment of GMP and the cell culture lab, and another on the training you want for the advanced techniques of vaccine production. So we can schedule it next financial year. We'll take it, put it in the calendar as well, and you can tell us how many of your trainees you would be sending, because here it will be requiring some hands-on training also. So I think virtual you will be able to do it. Yes. So I think this can be taken up very easily. You have any questions? Yeah.
So just because I am closely working with this Institute of Veterinary Biologicals, they have infrastructure. Infrastructure is okay. Thoughts are also visited, but definitely to clear the GMP, some modification will be required. Second thing about the instrument. Nowadays, every time it will directed to go to the SAM, but to get a quality instrument, otherwise vaccine, lyophilization, everywhere it will suffer. So how to put some rider that for biological at least, this quality of instrument, this criteria should fulfill, then only they should purchase. Regarding instrument, because instrument will be the main things to produce the quality vaccine. Second is the manpower in the State Department. All the director, I think they will agree. The trained person, they will be transferring immediately. Some central policy or some suggestion can be given. At least trained person should retain minimum 5 years, so that next batch should be trained up. Then only he or she can be transferred. This type of policy, if we do not put there, this production, biological and vaccine production, will always suffer. These are the issues what I observed here.
Regarding procurement through JAM portal, there should not be an issue. Now there are a lot of options available whereby you can choose these specifications according to your requirement by developing the quotation within JAM portal. So with higher specifications, you can easily eliminate those unscrupulous vendors who supply low quality equipment. I think there are some ways and means to be followed, but of course that needs some expertise to the group which is involved in procurement.
For
Otherwise, it's
procurement
possible.
of these things, I think
It's possible.
if they will put the institute or experienced person as in their committee member, I think that way they can assist this.
But we cannot skip that now even within the university has made it compulsory to have all equipments purchased through their portal. So I think that we must have to follow. The government instruction has to be followed. But we have to try for finding some ways and means so that we can skip or eliminate those unscrupulous people or vendors who do not supply quality equipment.
One thing can be done that there should be committee members from the institute who is knowledgeable about the equipment for preparing specifications. The state government there not be that much experience. So Dr. Verma or other experienced person can be there in the committee so that they can select good equipment.
Right from the
Beginning.
yeah.
So the best procedure is to go for a BOQ build. So BOQ build is a customized build. So you make your customized build according to certain features which are going to eliminate all these second or third grade players. And then you can go for your preferred brand. But it's mandatory that whoever is selling a good equipment has to come into the GEM portal. So that is also, but GEM can't be avoided at any cost.
Okay, thank you. I think from Assam we have the inputs. Now we move to Arunachal Pradesh. So we have the director.
Most respected Honorable Director, IBR, Madam Literacy, Mr. Bora sir, Brahma sir, Mr. Kutsam sir, and all my colleague respected directors of the United States and scientists and heads of various divisions and officers who have accompanied the directors from the states. Well, it has been a very fortunate occasion to be here in this interface meet with the Honorable Director and scientists, senior scientists, regarding the issues in the livestock, in the health and the livestock development, and particularly the emerging diseases which we are facing, particularly in the northeastern region. We have, everyone knows we have experienced in the last 2-3 years many emerging diseases outbreak has taken, and how we face it, we all experience. And it's fortunate enough that today the IVR have come all the way here in our northeast and apprise us, explain us the status about technologies being developed, especially in the veterinary biologicals, that is vaccine development. What are the vaccines have been developed and what are in the stage of developing, and how long we'll have to wait for the vaccines like LSD and ASF, etc. Now, just now, our respected and then Mormon sir has explained about the situation and the status of the emerging diseases and how we are tackling and what type of vaccines are being available in the states. He has vividly explained, meticulously explained about, and also about the center, the regional center at Kolkata, which needs lot of assistance, lot of hand holdings from the IVRS. I have seen here in the program, IVR has conducted training and other programs last in 2016. Then after, I think such program has never taken place. So my view is it is too long gap. There should be such programs should be held frequently, if possible, in every state, so that the local veterinarians, the local staff, they will be morally strengthened. Then they come to know that from IVRI, the scientist has come here to learn our problems and also to give us the solutions. That way they will have their moral spirit will be very high. Otherwise, as you know, in the states, particularly in my states, Himachal Pradesh, so many institutions, especially concerning disease diagnosis, like EIS, DDLs, those were established in the 80s and 90s, and their conditions are very deteriorated. Not to talk of modern instruments, even the building structures are totally dilapidated. And then we are facing skilled, trained manpower also doing the diagnostic activities. So I wish that IVRI should make some sort of plan so that they can come to the states and conduct some sort of training, awareness, hand holding, particularly in the case of prevention of these emerging diseases. As we have heard, that we have learned now, that many vaccines, they may not be available very soon. But take 1.5 years, 2 years, or maybe more. So in the meantime, if he can have some sort of plan, some sort of SOPs, for the farmers to rearing animals, mainly pigs, in our places. Even goats and poultry also. And especially in our Arunachal, in our case, we have those two very precious animal livestock producer between India. Our state is having the highest population, not only in India, but so having such a precious animal, if we are not serious, on the other hand, emerging diseases are day by day, public is taking place. So much precious animal. So, while planning such plans, IBRI took an innovative plan, how to be normal plans also be developed. National, as for the national base, if you do planning, that may not work. Those plans, those are the basis of this in some areas, especially in our country. We have the four climatic zones. So, larger areas of our country, almost 1 million from maybe two. We have very and the conditions, the situations, the locations, they are completely different. So, for such areas, at our state, different kind of innovative plans, SOPs, advice from IBM, it will be very, very grateful. And also, our present condition of our diagnostic lab, we have around 15 labs in the district level. And zone level, we have 4. Those labs are also in the hand of IBRI, it can strengthen, it can improve not only the infrastructure facilities, but even the men who are meant those they are also trained and holding great help. There are many things to say about them.
Thank you.
Thank you for putting up a number of issues related to Arunachal Pradesh, and you are very right. In 2016, you organized the interface meet. I think after that, maybe in after that, I think this is the first time we are having an interaction. And surely we would be like to have some programs from our NEH. We have the NEH funds. So specifically, I think we can plan something in Arunachal Pradesh, especially as Sir has rightly pointed out, some training programs. And we are in the process of developing package of practice, which you told for the farmers. So I think we have some people here who are into it. We are in the process of that. So we will put a separate part for the NEH region, and we can take in collaboration of NRC, YAC and NRC Mithun, so that we can develop such package of practice specifically required for your region. So we are into that process in this financial year. We are like making it. So if possible, we will include the suggestions very rightly pointed out by you. And apart from that, whatever you have told about organizing trainings and all, we will see how we can collaborate with you in the NEH program. We have the NEH program, and in that we are going to various states for organizing capacity building, awareness programs, goshtis. Our this ERS Kolkata team is actually having the whole funds of NEH. So I think we can plan something and going there. And apart from that, empowering your veterinarians that we can do through trainings. You can depute some of them for a specific training. You can write to us for a specific training program which you would like to. And interface meet, of course, we can come to your place, like we have organized it here. We can now take it up for separate states according to their objectives and needs, so that we can do. So, sir, thank you very much. Very innovative and good ideas provided by you. Now, moving forward, we have representatives from Meghalaya, Dr. Priya Joshi, Joint Director and Registrar State Veterinary Council, and Dr. H.P. Paul, Deputy Director, Meghalaya. So, whether you have any input, sir? Yeah.
Good afternoon to you all, and with due respect and our guest, today's Director of IBRI, Dr. Triveni Datta, Joint Director Extension, Dr. Vipasi Tiwari, Dr. Pramod Gora, and all senior scientists of IBRI and the representatives of all the different northeastern states and all officers present here. Yeah, today's session interface meet was very knowledgeable, and we are really greatly profited by this one. So regarding Meghalaya, we have got some specific points. I will be going to the specific points only. The first point we are in very much need is establishment of ICR Nibedi Regional Center at Umiam, Meghalaya for containment of transboundary exotic livestock diseases. Meghalaya is dependent on animal husbandry for livelihood and nutritional security. The state is having a long porous international border and is gateway of other northeastern states like Mizoram, Tripura, Manipur, and southern Assam. ICR Nibedi will be a befitting institute to handle issues of exotic livestock transboundary diseases, research on emerging and re-emerging livestock diseases. The existing resources of ICR RCNES may be utilized for the immediate setting up of and operationalization of this center. That is my first point. And number two, we have got the Animal Husbandry and Veterinary Department. Government of Meghalaya is planning to establish a veterinary college in Ribhoi district in Meghalaya, and we want a technical collaboration from the IBRI and ICR Institute for that purpose. And that is, and also we are planning to establish one dairy science college in Tura, also West Garo Hills. So we want a technical collaboration and support from IBRI and ICR. Number 3 point, the point number 3 we have got is increase in the number of state quota in CBSC Aizol and Jalupi. In CBSC Aizol we have got only 9 number of seats allotted to Meghalaya, and Jalupi we have got only 3 number of seats. So we want it to be increased by 2 or 3 numbers of seats, because in Khanapada we have got only 2 seats now instead of 4 last, no? And we also want some training for organic livestock farming. So for this, IBRI is a pioneer on this field, and we really want off-campus training. Will be a priority for us, because to send the farmers to IBRI also will be very tough for us. So these are the most important things we'd like to put. Plus the issue of ASF is common for all, so that also may kindly be looked into. So with these things, I would like to end here, because many other speakers are also there. So I will.
Thank you, sir. Most of your issues are related to a number of policy decisions. Of course, IBRI can put a word into the platforms where the policy decisions are being made. Of course, one of your points regarding the training for organic livestock farming for an online mode, that can be taken up. We can do that. So I think, of course, we are having the proceedings, we will be preparing for this. But if one letter can come from you to our IBRI, this can be easily prepared and implemented in the next financial. Thank you very much.
Thank you.
Moving ahead to now, we have the state of Nagaland. So we have a representative from Nagaland.
Before that, we have Mizoram.
Mizoram. Oh, sorry, I missed Mizoram. Yeah. So we have Mizoram, we have Dr. Director Animal Husbandry. Yes, sir, please.
Okay, thank you, ma'am. Respected Director from Vice Chancellor, IVRI, Dr. Trivedi, sir, and all other senior scientists from IVRI, my colleague and representatives, directors from our sister states, and it's a interface meeting is a very unique, I should say, because instead of this interface meeting, I should say it is something like a refresher meeting. Because the knowledge which we have already acquired that we're inactive and dormant are present today through your presentations. It's very encouraging and refreshing meeting today. First of all, during the repeated RCM meeting under this ICL, which we have through virtual mode, from the department, we used to have certain points and issues that has been marked from the authority. First of all, establishment of this small biological unit for area-specific requirement. Actually, there is a little bit history. It was in early 2010s or bit earlier than that. There was an outbreak, I should say, incidence of classical swine fever in various parts of Mizoram. Those days, the department inquired and seek the availability of viable classical swine fever vaccine across the country, including Assam. But it was not available at all. And there was an acute shortage, and in the meantime, there was a very high demand from the public that there was a great loss because of this CSL. And the state government, as well as the department, proposes to include this establishment of this biological unit based on the specific needs, particularly the classical swine fever. But time has passed, and as we all know today, that the central government is formulating certain policies and guidelines that these vaccines distribution as well as supply to all the states are centralized. And I think we have learned a lot from this vaccine production that has been transferred, technology transferred to this company now. And from this central government guidelines, the requirement based on the what do you call this, the population has been chalked out for the respective states. And based on that requirement, the central government has supplied all the required vaccines, including this CSL. And the issue those days has been over at present in respect to this establishment of biological unit for production of classical swine fever from Mizoram. I think this issue may be kept in abeyance from today onwards, please. That is the first issue. And the second one is regarding the training needs and training requirements. We have gone through all your training manuals and modules from the IVRI which are downloaded. And we have gone through all the topics, postgraduate course, diploma, vocational course, and short course training and all. And based on the requirement of this training protocols, I think we may be able to avail it in the days to come. Over and above these training protocols, which we have already scheduled, the department is establishing a BSL2 laboratory at present. And we are in a final stage, and installation of the equipment is underway, under completion, and we expect to complete it in a very short time, if things goes right. Of course, we may have these facilities, but to make use of it, we'll have to have a technical expertise. And for this, running the BSL2, in consultation with your esteemed research center, I think we may be able to send our officials to have some sort of training irrespective of the handling as well as management of all these things. Maybe PCR or maybe reverse transportation and everything. I think we may be in touch with the authority so that we can send and train the officers for running the PSL too. That is one request. And the other thing is that in regard to African soil fever, that has been already explained, as Professor have already said that there was a huge economic loss due to this African soil fever from Mizoram, as we all know. And I think they're proactive people that they have learned either from this internet, or there is a viable vaccine from Vietnam and it can be procured legally or illegally, I do not know. But I have learned that some people have already procured and they did it. And we discouraged so much that it was not continued. But it was unofficial, of course. And we have learned the things, the reality of this vaccine produced from the Vietnam that it is very interesting, as well as it is very good to have that knowledge so that we can answer to those people. It is very questionable at present. In the meantime, we are eagerly waiting for the production of viable vaccine, but the complexity of the vaccine production that we all know, but let us wait and let us hope so that we can have at least some measures so that people may get benefited out of it. And lastly, ultrasonography as well as X-ray imaging. This means imaging technique. I think it is very important for our diagnostic, which we are using in today's activities, like maybe pregnancy diagnosis as well. And this kind of training, I think IVRI may be able to extend the technical support so that we can make full utilize of those small machines, other handling, technical thing, as well as interpretation. I think somehow, somewhere we can do, of course, but not to that extent, so that we can make a maximum use of it. I think I will not extend much about my speech because time has passed. And with this, I conclude my remarks. Thank you.
Thank you, sir. Of course, from now on, because this was going on, the establishment of biological production unit in the regional committee meeting 3 for many years. So now after this meeting, I think now we can write to them that this may be kept in awareness. This is not much required now. And for the training needs, of course, these things we can take up. That is the handling and the management of BSL2 labs, and of course the imaging techniques. These are already there in our the brochure you have shown. So I think you can nominate your officers for that, and even you can write one letter to us for customized training. The things that are not there, even that can be written to us, and we can design and customize the training for you, especially as per the needs of your department. Now moving ahead to the representatives from Nagaland. Nagaland, we have Dr. Keduzul, Deputy Director, Nagaland. Nagaland, yes.
Thank you for that. Chairman of the second session, Madam Protosi, Madam, respected Director, Sir Jubilee, Promoter, Sir, 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 Directors of the sister state, Promoter, and then all the scientists present here. I will not take much time, but before I leave for this meeting, I met with this director and traditions. So I will present all a little 2 points for the house. It is really highly encouraging to see all the development in the veterinary field. I believe that our country has developed a world-class laboratories. So it is really encouraging. In a very short time, it has seen that LSD has been developed. It's very market. So within a short duration, all these things has been happening. So I believe that the technology we have developed on very high. Coming back to field levels, our director has suggested some point. Learning from the TAD disease outbreaks in the Northeastern state, such as reconstruction, is one of the main issues in all the states. Then coming back to this LSD, African swine fever, when it first outbreaks during 2022 in March, it takes us at least 3 months to get the confirmatory diagnosis. Then in the subsequent outbreaks also, since we don't have the facility in our state to diagnose such diseases, so preliminary diagnosis also cannot be done at our own level. So there is a request from our state that certain empowerment of the state laboratories, especially the one central lab, should be developed in the state. Then secondly, some of the economic important diseases also, like we have very limited resources to carry out the diagnosis. So the suggestions which our madam has put forward is to develop certain rapid diagnostic kits which can be used in the field level to rapidly diagnose, so that the epidemic impact can be controlled or prevented at an early date without much spreading. So that was the suggestion. So I, on behalf of my state, request the research community to focus on certain economical important diseases on rapid diagnostic kits. Then there is one point again. The second point is especially nowadays the dairy farmers, they are very much enthusiastic and very keen to see the pregnancy status of their animal. So unlike this medical, we don't have a rapid pregnancy diagnostic kit. So that also has been suggested by my director to put before you to find out some solutions for developing pregnancy diagnosis. So with these two suggestions from my director side and from the state of Nagaland, and I thank our announcer for organizing such a beautiful means to highlight the piece of our veterinary science, especially in the Northeast. So thank you so much. Then lastly, I support the advice or the points put forward by our director from Ireland. Thank you.
Thank you, sir. We have taken note of your suggestions for rapid diagnostic kits development and the pregnancy diagnostic kit. Actually, pregnancy diagnosis kit, it's already developed. One kit is developed by CARB. So I think they are in the process of commercialization. Commercialized?
It has been developed, ma'am, and it is in process of commercialization.
Commercialization. So I think soon you will be getting that. It will be available in the market. So that is it. Thank you. Now we move ahead to Sikkim. We have Director Animal Husbandry.
Good afternoon once again, Director of PCC. My professor, Dr. Golani, I used to be a student. You tend to forget faces, I know that. There are thousands of students that he has trained and taught us. And and all the senior scientists, directors from Northeast. Well, the difficult part is when all the intellectuals finish talking, when you are coming in the bottom and you have to say, you have to repeat the same thing once again. So that is sad. Despite that, Sikkim, as you all know, is an organic state. And we were also, hammered more by LSD and ERS. And we have to depend on, getting the vaccines from outside the state. We do not have that facility, so, because our population is also very small. So we always look out for the best vaccines available or best treatment methodologies that is provided by, institutions like, So we are completely dependent on whatever is produced outside. But side by side, being an organic state, we have to look out for practices. We have just got a roadmap of how to be organic in our state. And we are at this juncture, we are focusing more on ethno-veterinary practices. And if IVRI can also suggest us on area of focus where ethno-veterinary practices can be taken up, so that we can send our veterinarians to these places so that they can learn and come back with practices. Though we've tried for a few places, we had some resources in Tamil Nadu and Kerala and even Madhya Pradesh. So we invited them and then we had 2-3 sessions of training. But I think that is not sufficient and we have to move forward. Likewise,
I can give you the contact number. One Dr. Pankaj Doctor working in the NDT.
Fine.
He is working with that line.
I see.
Particularly in therapeutic line, he's developing some ethno-veterinary research.
I see. That would be very nice.
In NDT. So I can give you.
Fine, sir. Fine, sir. That would be very nice of you. So that
you are also our student. That's why.
Okay. So we can have an interaction session also. So this is one area I think we should need to focus in regards to Sikkim. Another is I'm very thankful to IVRI for your activities that is being carried out in Sikkim with KVK. I got to know, I was just going through this document and most of the news are very viral. That's what I could understand. Anyway, I think we have to have more of this type, and let us also coordinate with the Department of Animal Husbandry, because KVK is doing their thing separately, so that this knowledge sharing process, if it is also conversed with the Animal Husbandry Department, I think it could work more on it. So that is one area of intervention that I think we should need to initiate. It was, I was a little surprised when I came to know about 75% decrease in population of donkey in India. So I think we have to, as a veterinarian, I think we need to work more on propagating and conserving these type of animals. I remember we had in our place only we had a pure breed of pigs. So what happened all of a sudden was it's almost vanished now. We cannot bring it back. But that type of breed is available in Nepal. It's a pig breed, basically. So conservation and propagation of such type of breed, if ICR and IVRI can, with NBLGR, can initiate programs on conserving these type of breeds, would be more encouraging for. In terms of trainings, we also our para-veterinarian, who are the main backbone and a link between a farmer and a organization. I think we have already listed out the list of trainings that are required. I think that was read out by Madam also. So I think these, all the, trainings that are required for veterinarians, even for knowledge sharing with farmers, awareness camps, all this have been already listed out. So if this, just doesn't become a document, and if some areas can be captured, it would be very nice. And I also would like to suggest this organization being completely technical, but I think we also need to focus more on, economically uplifting our farmers where, these areas can be focused. So that is all the suggestions I have and requests as well. And thank you for inviting us in this platform. We would like to meet once again if you are invited or wherever you call us. Thank you so much.
Thank you, sir. Your points are noted, please. Ethno-veterinary practices you have suggested, so work is going on in IVRI, and we have here, I think, the head of medicine division. If he wants to provide any inputs, else we will provide you the number. Dr. Akhilesh is the coordinator of this meeting also, and he's also working on veterinary medicine. So you can link to them, and we can have the trainings whenever you are requiring them. Apart from that, you are talking about conserving indigenous breeds. So IVRA has taken up a project with NVGR, but we are working in the Uttar Pradesh area. But of course, if you can write to us or to NVGR or the breeds that you are thinking that they are depleting, then we can take up. NVGR can take up with, I think, maybe AU, Hanapara, or any other research institute in the area. So that can be done very easily. And NH program is already there. We are doing it, but mostly with the KVKs. I think we can collaborate with the animal husbandry department, and that can be very effective. Dr. Indra Subba, she's our alumni, and she has provided them great help. And she's always the link. I think we can link to her and then organize more programs. So thank you very much. Since we are moving very fast because we are losing time and we are reaching to the lunch time. So we have the next state is state of Tripura.
Good afternoon, Honorable Director, Vice Chancellor, IVRI, Dr. Triveni Sir, Dr. Rupasi Tiwari, Dr. Bora Sir, Dr. Pranab Sir, and most familiar face to the beds of Tripura, Professor N.N. Burman Sir. He visited several times to our state. And all the participants, scientists in this interface meeting. It is a great occasion to meet with the top officials of IVRI, that doing interface meet, especially for the northeastern states. In the very beginning, I congratulate the organizer and our director, ICR, IVRI, Ijjatnagar, to consider the northeastern states as one of the important sector for developing their next plan, next program. And they have called all the related officers, director from the concerned directorate of the states of northeastern region of India. As we are from northeastern states, very close to each other, our issues and our proposal also may be same. I request our madam not to avoid anything, because this may be repetition of the proposals. In Tripura, Animal Resource Development Department has started one project named as MSPY, Mukhyamantri Swaনির্ভর Parivar Yojana, right from 2018. It's a very popular project. And in this project, department as well as government has decided to touch each and every family of the panchayat level, village committee level, through supplying of the inputs, different inputs, like poultry, like duck, like goat, like pigs, etc. Those who were rearing the animals and birds from the beginning, other than this, the beneficiaries are included in this project. While implementing the scheme in the field, we found there are some gaps. Gaps in this sense. Our officers, our doctors, they are very well trained from the institution where they got their degrees, master degrees, etc. But very recently, so many emerging diseases have come out, and they have very negative impact on the earning of the beneficiaries or the farmers. I can tell here, swine fever, African swine fever, LSD, etc. From the morning right now, it has been discussed in various ways several times. And Tripura is not far from this effect. Our doctors are working in the field. Our ARDS staff, that is technical staff, they are also working in the field. You all know one project is there, Rashtriya Gokul Mission, RGM. In that project, unemployed youths were selected and they were trained as the doorstep AI worker in the field. They were at us. Lastly, self-help group people, self-SG, the team leader of that SSG, that is the Poshu Shokhi, to maintain the vaccination, treatment, etc. of those distributed inputs. In this sense, there are so many farmers, they have lost their pigs, they have lost their cattle. We are trying, our officers are trying in the field level to recover from that situation. But basis on this, it is required further training or refresher training under capacity building project. And this will be done from the premium institution, IBR. That will give a very, very good impact on the officers, the staff who are working in the field. Because they will come directly in touch with the IBRI. As well as there are 4 labs, disease investigation lab in Tripura now working. There are 8 districts, 4 labs are there. Officers working there, they also need different training on laboratory, saving the life of the animal through treatment starts with the laboratory findings. So if that sector will be developed much, it will be really helpful for the farmers, and experience will be gained to the doctors, and all the projects in all the states, I believe, will get a success. Lastly, another proposal. Already it is matching with so many states. Our state is procuring vaccine from outside state. As Tripura is in the end point of India, vaccines are being carried from other state. It takes 3 days, 4 days, 5 days like this only by transportation. Sometimes there is acute shortage of vaccines in the field. We do not get vaccines. There are huge crises. Farmers, they come to us, they put their demand, and that is being discussed. That the government is not in a position to supply the vaccine. To avoid this situation in future, our humble request, if at least a small unit of vaccine production center can be taken into consideration at Agartala at least, or at least in the college. Veterinary college is also there, only one college in Agartala. This will help in future also. I request to the authorities of IVRI, please consider. And lastly, I refer to Dr. Bora sir. While we were in the service at the very beginning, we found so many officers, senior officers, they are trained from IVRI, diploma, etc. So many days after their retirement, now nobody is there trained from IVRI. So I request to the authority of IVRI, please start once again, at least 2% or 3% from the state to give the training in diploma or anything. It will be a very good step to develop the veterinary sector in Tripura, in far east from New Delhi. At last, it's really a very good experience for us, for this gathering. And so many scientists, Thor sir, Mormon sir, and Johan sir, they have given the presentation. The latest thing, I think the latest of 2024, this is the February only, we have seen in the display board. I am very much grateful personally to them. And this will continue, I think. This type of meeting will encourage us to tell you our demands, which I expect it will be successful in future. Thank you, sir.
Thank you, sir, for training, refresher trainings, for disease investigation lab issues, and for the veterinary officer. I think we already have a list and brochure. Yeah, I'm coming to this. So we are already having that brochure, and I think you can write to us about specific training, or you can nominate for the as per the training calendar. That is the old training calendar. The new one we are in the process of making. So the 24-25 calendar will be soon available on the website, and you can write to us. Apart from that, the diploma for the field veterinarians, we were having a large number of diploma courses. So we have the joint veterinary academic. So we will take his inputs after we listen to Director Manipur, who is awaiting to give his points. So now I request Director Maninu of Tribotumbi to please provide his
Yeah.
inputs.
Good afternoon, everybody.
Yes, sir, please.
Can you hear me?
Yes, yes.
Yes, sir, we can hear you.
Good evening, everybody. Good afternoon, everybody. Actually, I'm very sorry that I could not present there because of the, coinciding meeting at Delhi also. And two of our officers have been sent there for the ensuing this livestock census. There is a meeting at Delhi. So I could not come there also. And first of all, I extend my regards especially to the director, come Vice Chancellor of ICR, IBR, Nagar, as well as Dr. Arnab Senji for holding such a important meeting of interface, meeting with the northern states. And I extend my regards to all the renowned scientists of IBRI, as well as all the northern state representatives and directors of any states. And actually, first of all, I would like to extend my gratitude and thankfulness for convening this interface meeting, especially with the IBRI, with our northern states, which is very important for us. And I've been here itself since the beginning of this meeting. And already, most of our state departments, representatives from state departments, they have already expressed their views regarding the current requirement or present requirement for various training, then scientific knowledge and diagnostics, all these things. And for me also, regarding our state also, I have already shared some of the points or topics which are practically important for our states and northern states also. Just for naming all these things. Number one, capacity building for the field veterinarians and the paravets. Their trainings regarding our recent scientific knowledge and the techniques in our animal husbandry in a field condition is highly required. And moreover, I have already participated such interface meeting in the year 2000, in last year in month of September, and the training which are short-term training courses, then other different courses which are being conducted from time to time by IBRI. I have already received it, but for our state, due to the current this law and order situation and the last African swine fever outbreak, which has been extended in 7 districts, that is widespread. To control containment all these things, our field veterinarians or officers, they could not be sent out. But in the coming years, we'll be sending them to the IBRI for required training programs, what we presently needed in our state. So as it was given in the discussion already regarding the conservation and the development of certain indigenous or non-local, non-descript local breeds of our northern states, like cattle, this buffalo in Manipur also, there is our local cattle is there, and this our swamp buffalo is there also, and some of the poultry breeds, and there is one dog which is called Hafa in the Ukhrul district, which is having its unique, features, characters, which are very much suited for the hunting. That we would like to conduct, or we like to submit some of the proposal for characterization and study through IBRI or through IBJs regarding the establishment of these breeds. And it is also included, I think. Then another one regarding the animal disease diagnostic, different, these latest technologies in the animal disease diagnosis and at the molecular level also, we are very much highly needed. In Manipur, there are earlier 9 districts were there in our state, and due to the further further development of our state, there are 16 districts have been developed. So as our plan, we are going to set up 9 district level district laboratories. So far we have 6 only. So these manpowers have to be trained for the different livestock disease diagnosis. As some of them, what we learn it, what we know it, we are also giving, we are imparting training to them. But especially the new technology in the animal disease diagnosis, we want to send to the IBRI, and the latest technologies which are very much required now, it's radiotherapy, ultrasonography, all these things, and different technique in surgery also. We like to send our officers, field officers to the IBRI from time to time. Then another one regarding the availability of diagnostic kits. Earlier also we are depending some of the kits developed by IBRI. As it is very less costlier compared to the products which have been marketed by other private companies. So most of many of these diagnostic kits we are still required, and we like to purchase it through our state fund as well as SCAT. That's why I would be very much thankful if you kindly give me the list of the this available diagnostic kits for animal disease diagnosis and poultry diseases also. Then regarding the another training parts in Manipur, actually under RGM, Rashtra Gokul Mission, we are also giving training for the AI to the Maitris, as Tripura director has already mentioned, and this Pasu Sakis like that. Same thing, we are also giving it. But some of this AI technicians, we like to send to these northern states either at Kolkata or at Siliguri or at IBRI for learning the AI technique in sheep and goat and pigs, because these are also very much important for our state. Then regarding as it is also included in a meeting, regarding the study of the transboundary diseases, it is highly needed in our state because Manipur, it is at the boundary state of India.
Sir, connectivity is failed. So we cannot listen to you.
Yeah, Dr. Ikotomi, could you please reconnect because your link is lost apparently.
Till the concern comes back, so I think we can move ahead. And we have our IBRI center with us. So if we have any specific points, especially we have Veterinary Academic with us. So sir can tell us about the diploma courses and how the rejuvenation of the diploma courses, because we right now I think we are running only one diploma, national diploma. So sir will tell more about it. So Dr. S.K. Mediratta, Joint Director Academic, please.
Thank you, madam. So, Factory Director, sir, Joint Director Extension, Madam Roopsi, Dr. Borad, Dr. Burman, Directors, Deputy Directors, and Officers of Department of Animal Husbandry of different states. So, as highlighted by different directors and joint directors, so already we are running number of diploma, certificate courses, and vocational courses. And I am thankful to all the directors of northeastern states that we are getting sizable number of participants for these diploma and certificate courses. So in last year, we have relaxed conditions. So earlier there was maximum 2 participants from one state. So last year we have relaxed that condition, and we have also relaxed the age condition. So that is why we got more number of participants from these states. So I'm really thankful to all the officers for nominating veterinarian for these courses. So one demand that the diploma, those who are coming for the diploma, so last year also they were asking that why we were not being sponsored for this PG degree. So today is the right time, so we can debate on that. So earlier we were having this PG sponsored program only for these persons who are serving in state agriculture university. But with the change in time, if you feel that field veterinarians should also be trained in postgraduate, they should also be having postgraduate degree. So then we can put up this agenda in our academic council, and we can also get approval so that state veterinary officers can also be sponsored for the PG program. So if you feel, you can suggest to us so that we can take up this matter further. So because if they will be getting 1 year this coursework here, and then research work they can do in NRC Mithun, NRC Yak, NRC Pig, or even Assam Agriculture University, or wherever some other this research institute is there. So they can 1 year they can spend in IVRI, and 1 year they can spend in the northeastern state for research work. So we can debate on that. So, and we will be happy to introduce new training program. So whatever that training or this capsule program we will propose, we will be happy to take that. So once again, I want to thank all the officers and giving me the opportunity to speak. Thank you very much.
Thank you, sir. Do we have any other inputs from the IVRI Izatnagar Center? Any other head of the division would like to put out some points? Okay, if there are no points, then we will be winding up this session. I have my co-chairperson, Dr. Bablu, with here. So, do you have to put up some points?
That madam, if that Ivotombi he communicated something over phone, he another point that all the bacterial vaccine, anthrax, XL, BQ, since Assam veterinary biological they are producing, they should strengthen so that they can supply to Manipur or other northeast state also.
Strengthening the Assam biological production.
Yeah, production size should increase so that they can also procure directly from the Assam.
So if Assam government can send one letter to us for this hand holding and the strengthening, then we can initiate the process. Thank you. Dr. Gupta, please.
Thank you, ma'am. Most of the things have already been discussed. I just wanted to make one point that we are running a project on promoting entrepreneurship and startup. So whoever is from university, I would just like to request that the students, the innovative student can participate in those programs, and we have a funding opportunity also for 25 lakhs. So it is a grant money. So those students who are innovative, they can take up some small project in collaboration, and they can participate in this. And we have also several entrepreneurship development program. We can also formulate as per need also from the states. Thank you very much.
Thank you, Dr. Bablu. I would like to add up here that apart from students, farmers, entrepreneurs, anybody who is interested to run a start an enterprise, they can just send their application to Dr. Bablu or to us to direct to any mail concern. And whenever we are advertising our cohorts, we are advertising our batches for seeking funding. So we can send, if there is a group, we can make a WhatsApp group of at least the representatives here or the Director Advisory of any states. We do have one of all India State Advisory Department, but if any state specific, we can make it. So we can post our advertisements there, and you can nominate your people. They can come. They can appear online. We have many courses of entrepreneurship development that are online courses. They do not need to come. So there are many issues like developing startups and entrepreneurs for pig farming and many other things of various states. So I think that can be served through these two programs of agribusiness incubation and Raftar agribusiness incubation for startup and entrepreneurship development.
Take the opportunity to share another information on behalf of our university. I think many of you may not be knowing that since last August 2023, the Assam University has also started one ODL platform, open and distance learning platform for entrepreneurs, farmers, rural women, and any stakeholder in the agricultural sector. So there are courses, total 58 courses have been identified, already is being launched. Under that, there are courses in agriculture, veterinary and animal husbandry, fisheries, horticulture, sericulture, and others, including home science and community science. So any farmer or entrepreneur, or even the para-veterinary staff may also take some of the courses that are delivered mostly 80% online. They need not have to come, only towards the end of every course. They should come to the campus from where it is being provided. So for maybe say about 3 to 7 days maximum for getting the practical training. And there are 3 levels of courses. Some courses are short duration courses of 14 days to 1 month duration. They are simply say short-term training courses. There are courses which are from 3 to 6 months, certificate courses, and also there are diploma courses 6 months to 1 year. So now we are working with only the short-term courses and the certificate courses. Already 22 programs have been over, and there are participants across the northeastern region. I have seen people from Tripura, people from Nagaland and Arunachal Pradesh. Some of the people have already participated, but we can popularize them among the farmers, entrepreneurs, and also the veterinarians and paraveterinarians, even people from various backgrounds, like some advocates, engineers, doctors. They are also joining the program because many people are nowadays interested to take on the agriculture and allied sciences as maybe alternative means of livelihood or subsidiary income generation like that. So that can be popularized because this is delivered in both English and Assamese. So as part of your choice, you can take part in that. And fee is very, very minimum. Very, very minimum. For a 6 months course, it will be around 7,000. For a short-term course, it will be only 500 rupees to 1,000 rupees. And it is a pride on the part of AAU that it is for the first time in the entire country that kind of courses are being launched, and it is being held by everyone. And also, there are courses already taken by the KVKs under Atari. They have proposed us to offer similar kind of courses to the scientists of Atari, and they are providing total funding support to such kind of courses. To the tune of 40 lakhs, they are ready to spend for providing training to the scientists and workers involved in KVKs under Atari. So that has been launched. So I think this platform can be utilized so that the people can get the training by sitting in their own home. Only for a brief period they need to come for getting the practical training. Thank you very much.
Thank you, sir, for your nice inputs and about telling about the ODL platforms. I think this will be really helpful for many people. So now we are moving to the end of this program, and I now request Director sir for his closing remarks.
SC, ST, OBC उनको बड़ी भागीदारी मिली कांग्रेस
Thank you very much. Now we have come to the end of the program. So thanks for a wonderful discussions. We are really grateful to all of you, and my gratitude to Assam Agricultural University, College of Veterinary Science, Kolkata, and a couple of suggestions, points emerged out of meeting. The most important point is the collaborations in upcoming areas of research with the AAU Khanapara, and also the validation of some of the important technologies. So I think this collaborations is really required for not only for Khanapara but also for ERS. Certainly, we love to collaborate in various activities. Here I would like to request the officials of universities that the areas where we really collaborations are required. If these are known to us, then we certainly like to collaborate in these activities, not only through our Ajitnagar campus, but also our various through our various regional stations and campuses. We have pan-India presence. We have 3 well-established regional stations and 3 well-established campus. So whenever we required these collaborations, we would like to have the collaboration with the regional station campus. And second most important is the sandwich program. This is really a very good suggestions. I am very grateful to Dr. Prabodh Bhurani for giving this very nice suggestion. I think this is good for both of us, for capacity building of our students. And in tune of National Education Policy 2020, this is the activities also find a place. So we would like to certainly start this activity at the earliest. We have our joint Dean IVRE University with us, Dr. S.K. Vedanta. Kindly make a note that at least this sandwich programs AU Khanapara we should start at the earliest, because we have a MU in the area in various areas of research, academic and outreach activities. So I think we will not waste much time in our activity. This our official communication. I think right away we can start this program. And this is good for all of us and good for students also. And other important programs have been suggested by our esteemed director and joint directors of various states of the northeastern region. And I think we already had a note of it. We have made a proceeding. And whenever we come, I think next time then we will come with the ATI. So this we have already make a note of it. And the most important is the capacity building of the officials and other stakeholders. Because this is the initiative of the government of India that any institute who have nurtured with ample amount of money from since a long period, that the infrastructure should be open for the stakeholders. Now any research activities or teaching learning programs will be initiated in tune of the expectations of the state line department and also the other stakeholders like industry, entrepreneurs, So my submission to all of you that although we have a detailed our schedule of the training and we have already placed in our website. In addition to these training programs, if you have some suggestions on our training schedules, or you have any requirement of specific capsule-oriented training programs, kindly let us know. So that accordingly we can revise our training schedule and we can also include some of the important capsule-oriented programs as per your expectation. So this I think very important activity we can start immediately, and I think this is good for all of us. And now we will also try we have a you see 19 animal science institutes, and some of the important ICR institutes are located at our in these northern states. We try hard to have a greater networking with these institutes, with the state line departments. As far as this registration of breeds is concerned, I think our sister institute NBAGR have already launched a mission on these breed registrations of each and every state of the country. So I think this can be taken if any prototype or any germ plot, new germ plot or unique germ plot with unique capability or unique traits if having in any state, either this can be informed to us. We can call a formulated team to try and document that germ plot, or you can also recruit directly to Director NVAGR and also the officials of AU Khanapara. They can also facilitate in registration and documentation of the important germ plot available in the state. So this and other in continuation with the suggestion given by our Joint Director Academic and Dean IVRA Dean University Dr. S.K. Mehta, that let us start trying to sponsor our undergraduate professionals for the postgraduate studies. And here we have a very fantastic arrangement that the student can have a 1 year classes on this offline at the IVRA campus, and the remaining part of the thesis he can complete at our AU Khanapara. And also if he desired to complete remaining part of work with the industry, so we have an option that a student can also work with the industry for various type of research activities or various type of project formulations, evaluations, activities at the industry. So now in tune of the national education policies and BSMA of guideline of the NCR, number of freedom have been given to the student and also to the university. There is a lot of autonomy for the student and also for the faculty and university. This can also be taken in a fast forward mode in order to upgrade the knowledge of our UG professional working in the field. So this can also be started quickly. And as far as the prices of the technology is concerned, this is also a very welcome suggestion. And this certainly we will discuss with our officials. And I think this is the point we can also take a decision at the earliest to down the prices of our technologies, especially for NEH state biologicals. So these are some of the important points I think emerge. But above these points, in order to strengthen the linkages between the national institutes, I think this is the responsibility of all of us to come out with our suggestions, come out with our expectations with the institutes, so that institute can redesign the programs regarding the teaching, research, and outreach activity. So for greater collaborations, I think the communications among all of us is really required. So we hope that in future we'll have a greater collaborations in various activity in order to serve the livestock owner and other stakeholders in a very effective and promising way. And we'll also see how best whatever the suggestions given our esteemed director and joint director of the state will try to include in our research, teaching, and extension agenda. So this is from my side, and the end. I am really grateful to all the esteemed state directors of animal friendly of the various northeastern states, and I grateful to officials of AU Khanapara. Thanks to our IVRA team at ERS Villachia, and also the team of our Isatnagar campus, who are also our distinguished head of divisions and joint director colleagues. They are also made a point, and they are also sitting. And my thanks to all the in charges, regional stations and joint director of IVRI campus. They are also, I am looking, they are also online. So this is the, I think, not only this is the northeastern state, this is the agenda of IVRI, that we are outreaching our activities with each and every state. This is the expectations of the government, and especially the expectations of our honorable agriculture minister, that we must have a greater collaboration with the state department of agriculture. So this is from my side, and in the end, I am really grateful to all of you. Thank you very much.
So now I request Dr. P.S. Banerjee sir for his vote of thanks.
Thank you very much, Dr. Bablu. Firstly, I thank profusely to our director, sir, for delving into this idea of having an interface meet. And I hope this moment will prove to be a watershed moment for collaboration between IVRA and all the northeastern states Department of Animal Husbandry. And my thanks are also due to Dr. Robert Bora, the Director of Research of AAU, who gave a very good idea about English program, and thus the locals become vocal at the local level. I am also thankful to Dr. Burman, who helped us in many ways, apart from his presentation, everything. He helped us in many ways in organizing this function. I am thankful to all the joint directors of IVRI, Dr. Rupsi Tiwari, and who are joining us from different campuses for their presence in this interface meeting, and all the heads of the divisions, my colleagues, and lastly but most importantly,
the officials of different states and one of the department states who took too much pain to come over here. And it reminds me of the seven sisters who found at least two brothers today, one in the form of a Khanapara and the big brother IVRI. Hope this bondage will remain long. Thank you very much.
Thank
We request everyone to assemble for a group photograph outside.
And after that, lunch.
डेप्यूटी डायरेक्टर हो गए।